Okay, hi everybody. So welcome everyone to today's workshop discussion on quantum biology. I'd like to thank the conference organizers for inviting me to moderate this session. We are here to delve into the intriguing intersection of quantum mechanics and the intricate world of biology. Trying to read my slides too. Okay. So from the inner workings of photosynthesis to the navigation skills of migratory birds, quantum biology offers us a glimpse into the profound ways in which quantum phenomena influence living organisms. Before we begin with the panel, I will introduce myself. I'm Paige Derr, and I'll be moderating our discussion today. My education is from the University of Pennsylvania, where I earned a PhD in physics with a focus on biological physics. Following that, I worked as an imaging specialist at PerkinElmer with focus on detection and high-content instrumentation. And for the past decade, I've served as a scientist at the National Institutes of Health, specifically within the National Center for Advancing Translational Sciences. My research primarily revolves around tissue engineering, aimed to understand cell behavior within natural 3D environments. During my time at NCATS, I've also had the privilege of co-chairing the NIH Quantum Interest Group, facilitating discussions on quantum technologies in sensing, computing, chemistry, and biology. Notably, we hosted Dr. Hameroff as a guest speaker last fall, who presented a lecture to our group. If you're interested in joining our discussions, our group is open to the public and can be found on nih.gov or by searching your browser for NIH Quantum SIG. The SIG stands for Special Interest Group. So quantum biology has gained momentum in recent years, studying the way that life can take advantage of the properties of quantum mechanics to optimize efficiency and provide survival advantage. While the concept of quantum mechanics influencing biological processes dates back to early 20th century thinkers like Erwin Schrödinger, who pondered the role of quantum coherence in life, it wasn't until the latter half of the century that serious scientific inquiry began. The field began to grow in the latter half of the 20th century as scientists like Rudolf Marcus explored electron transfer and chemical reactions, providing insights into fundamental processes essential for life. However, it was in the early 21st century that quantum biology gained momentum, fueled by groundbreaking research into photosynthesis by visionaries like Graham Fleming and Greg Engel. Their discoveries illuminated the role of quantum coherence and energy transfer processes within photosynthetic complexes, sparking a renaissance in the field. Since then, quantum biology has grown steadily, with scientists uncovering a wealth of new insights into the quantum nature of life, from the discovery of quantum effects in enzyme reactions to the exploration of avian navigation systems. The field continues to expand, pushing the boundaries of our understanding. Our panel brings together experts, each with their own unique perspective on quantum biology. Together, we will explore groundbreaking research, discuss emerging theories, and examine the implications of quantum biology for fields ranging from medicine to fundamental research. So today we'll be hearing from Stuart Hameroff, who will start us off. He needs no introduction. He's a emeritus professor at University of Arizona. Then we will hear from Travis Craddock, associate professor at Nova Southeastern University. Albert Siryaporn, associate professor at UC Irvine. Nirosha Murugan, assistant professor at Wilfrid Laurier University. Manisha Patel, doctoral candidate at UC Santa Barbara. And Philip Kurian will round us out, principal investigator and director of Quantum Biology Laboratory at Howard University. So, Stuart, I'll go ahead and get started. Thank you very much.
Thank you, Paige. Thank you all for being here. Great crowd early morning. As you probably know, everything else is on the other side of the hotel. The plenaries are here. Let's start tomorrow. Anyway, we ready to go? No. He's going to switch it over. So I'm going to talk about quantum biology in terms of what we call the quantum underground, and that term was coined by Travis Craddock, actually, in a paper we wrote together. And I'll explain why. And I think it explains how we can avoid the problem of the brain is too warm, wet, and noisy. What? Oh, I'm looking at this one. Okay, I'm sorry. Okay, the quantum underground, where life defeats decoherence. And the picture is the simplest version of this. It's a beaker with oil and water, and oil and water don't mix. And that's actually very important in anesthesiology and medicine in general because it's a feature of solubility, and it determines where drugs go that we give. So as Paige mentioned, and I want to thank you for organizing this great session, Erwin Schrödinger, in his book, What is Life, suggested life's unitary oneness derived from quantum coherence, and that memory was stored in aperiodic crystal lattices. And I'm calling this quantum vitalism. There was a vitalism in the 19th century, which was either electromagnetic or some unknown force, and it was kind of shooed away when molecular biology came along. But Schrödinger brought in the possibility of quantum coherence. But everybody says that biology is too warm, wet, and noisy for functional effects in quantum systems easily perturbed by thermal decoherence. If you want to build a quantum computer in a laboratory, you have to do it at near absolute zero temperature because any jiggles from heat will disrupt it. So that seemed to be a killer for quantum biology. But is it really? Because biology is heterogeneous. It's not a monolithic slab of material that's warm, wet, and noisy. If we look at this picture of a cell, we see water, or we see empty spaces which would be filled with water and charges with various lattice proteins. And the two large cylinders are microtubules, and you can see the individual tubulins behind them. They are the structural, part of the structural cytoskeleton, and the other filaments are mostly actin. And still, it's mostly water, 70% water. Why this is important is that, in anesthesia at least, if we give a drug, we want to know where it will go. And we know that by its solubility in a particular phase or compartment. And here's an image of the solubility phases of a human body. And can you see my cursor? Yeah. You can? Okay. So here is polar, highly polar. This would be water out here. So this arrow here goes polarity, going this way. This is permittivity, and I think this is dielectric. And down here is nonpolar. And all these different solvents tell you where different solubility coefficients where drugs or molecules will go. So for example, most drugs are polar. They have charges, electrically charged. They're soluble in blood and water. They can be given intravenous, intramuscular, orally. They get absorbed. Everything winds up in the blood anyway, even inhalation. But these are very soluble in water, and they seek oppositely charged receptors on membranes with matching lock and key conformations. That's how they bind. So they would be out on the upper left here. But what about uncharged nonpolar molecules with complete outer electron shells, like the inert atomic gas xenon? It's a single atom. It's inert. It has a completed electron outer shell. It doesn't form any chemical bonds or ionic interactions. And it's an anesthetic. It's a perfectly good anesthetic, although it's a little bit weak and expensive. But it can be used and is a perfectly good anesthetic. Now, there were other nonpolar inert gases, which are anesthetic, which selectively block consciousness while other brain functions continue. Selectively block consciousness. They take away consciousness and very little else. We can have our patients unconscious, anesthetized, but their brain is working. We can do electrophysiology, for example, for spinal cord surgery. The physiologists stimulate the extremities. It goes through the spinal cord. We measure from the brain, or go the other way, motor responses, to make sure the spinal cord's working okay, and if the surgeons are operating there, and yet the patient's completely unconscious. The brain is active, the patient is unconscious. How does that happen? That's a big mystery still, but I think we're getting there, actually. Now, the anesthetic gases have varying chemical structures, but each has a nonpolar surface, shown here in gray, of complete electron outer shells, which bind by the same weak van der Waals quantum forces that xenon does. So you can see how many? 10 different anesthetics. I've used, I think, all of them, actually, in my long career in anesthesia, except xenon. I haven't actually used xenon. Anyway, but others have. And the way anesthetics bind is that they, if you look at the lower part on the left, 2 aromatic rings, which have completed electron outer shells also, actually a little bit more, which I'll come to in a second. The electrons in one repel the electrons in the other and create a dipole, and it's an induced dipole, and that causes a dipole in the original one. So you have 2 dipoles that are like little bar magnets. They attract and then oscillate in terahertz or petahertz. And then anesthesia on the right binds and disrupts those dipole oscillations by forming its own coupling, but in a way that doesn't go along for the ride with the oscillation. So we had a bunch of these anesthetics at the turn of the 20th century, a bunch of gases that could be anesthetics, all different chemical structures, but they all seem to do the same thing, but with different potencies. They all anesthetized all animals tested. And each one had a particular potency called a MAC, the minimal alveolar concentration required to anesthetize 50% of the animals or humans. And so 1 over MAC would be the potency. So scientists searched for a correlate of potency of these chemically disparate structures. And Meyer and Overton, working separately in Germany and England at the turn of the 20th century, plotted potency, which would be 1 over MAC. Here it's shown in MACs. So as we go lower towards methoxyfluorine, you become, it's more potent. So methoxyfluorine has a MAC of 0.25. You only need 1/4% to put a patient to sleep, or 50% of them. So obviously we give more because we want to make sure that the outliers are covered. And they plotted all of these and along this amazingly straight line over several orders of magnitude that showed that their potency was directly proportional to the solubility in a particular solvent, olive oil. So olive oil is an organic solvent. We all cook with it, and it's got aromatic rings. And if we look at where it falls on the solubility phases, this is the lower right corner. You can see the bing there, meant to imply consciousness. So this is where anesthetics bind. And if you look at the molecules, they're all derivatives of benzene, the aromatic rings. Dimethylbenzene, methylbenzene, and benzene. So that's where anesthetics will go, and that's where they act to block consciousness specifically. So this is organic chemistry, and I took organic back in college, and I actually liked it. It was a very interesting course, and the basic thing I want to tell you is that it's based on the benzene ring, six carbons with hydrogens, and there are 3 extra electrons, and they stick up and below the ring to form an electron cloud of delocalized electrons. So this is kind of a quantum piece of matter, of superposition, because all the electrons are distributed. So it first forms these individual orbitals, and then the rings, and then the electron cloud enveloping both. So if you had a bunch of these benzene in bulk, it's gasoline and it's explosive. But if you have them spatially arrayed where you can have interactions between rings, like in graphene or in proteins, you can have quantum effects. For example, quantum dipole oscillations, spin transfers, excitons, phonons, photons, fluorescence, phosphorescence, and superradiance. And Travis and Philip, I think, worked on the superradiance idea. And so the fluorescence means that if you excite a protein with an aromatic ring, like tryptophan, which is an indole ring, on the left in the blue, it's excited to a singlet excited state, and it stays in that state, percolating down, maybe changing a little bit, for a period of time, moving to the right to the green, where a lower energy photon is emitted and that's fluorescence. And you can record that, and you can measure the time between the blue lines and the green lines as the tryptophan fluorescence lifetime. And that is in terms of both time, but also distance, if you have a system where it can propagate. And Arad Kalra and others of us published this paper in ACS Central Science last year, where we had a microtubule. We hit it with UV light, and it fluoresced a short time later and emitted from tubulins further down the chain, and showed so there was both temporal duration and a physical traveling propagation. And our conclusions were, first of all, that microtubules were surprisingly efficient light harvesters. They actually soak up photons of all different wavelengths. The fluorescence lifetimes and excitations persist longer than classical Forster mechanisms, and thus are presumably quantum in nature. And the lifetimes and the distance were inhibited by two different types of anesthetic: etomidate, which is an IV soluble anesthetic, and isoflurane, which is a gas, although we used it in the liquid phase for this. So basically, we showed that these two anesthetics inhibit this quantum optical process in a microtubule at ambient temperature. A couple other things about these rings. Back in 2003, Ouyang and Auschalam at UC Santa Barbara used quantum dots, the big green and red things, interconnected by benzene rings. You can see the little hexagons in between them. And they put in spin, and the spin propagated through the system. And this quantum spin transfer through benzene rings was increased by temperature. So at least this form of quantum propagation was not inhibited by temperature. In fact, it needed temperature. It's also worth noting that that psychoactive molecules like dopamine and serotonin, the pleasure molecule and the mood molecule, are aromatic rings, and you can see the cloud around them. And serotonin has the indole ring. And the psychedelics, LSD, DMT, psilocybin, all have aromatic rings that similar, including the indole ring in DMT and psilocybin. And this seems to imply that this type of energy or this type of structure has something to do with consciousness. Now, recently it was discovered that psychedelics, which are known to act on 5-HT2A receptors, serotonin receptors, normally on the surface of the neuronal membrane, the pyramidal neurons, actually bind to 5-HT2A receptors, which are inside the pyramidal neurons in their soma and dendrites, associated with microtubule 1A. So they're getting very close to the microtubules, and you can see in this image the colored binding in the cell body itself, the red and the blue-green. So these receptors, and therefore the action of psychedelics, may be inside the neuron directly on microtubules. And that's actually fairly new. And I was kind of shocked by this because I've always been taught that 5-HT2A receptors are on the surface. And then I found, oh yeah, they're inside too. I said, well, isn't that kind of startling? Yeah, they didn't seem to care. It didn't seem to change anything, but I think it does. But anyway, so there are, these drugs can get in and activate the microtubules directly. Now, aromatic rings also form amphipathic molecules, where you have a ring, if you look on the left, the little hexagon with the lines, and a tail which has charges on the end, polar charges. So oil and water don't mix. So what happens is the nonpolar rings attract, they induce dipoles in the other one, and then they start to oscillate, and so they're kind of in the middle with their polar tails sticking out. This is how proteins fold with aromatic amino acids, and also how my cells form and other structures. And this may be a key to the origin of life in terms of my cells in the primordial soup. Aromatic rings also pervade lipid membranes and nucleic acids. In the lipid membranes, they bind together in the middle, and the polar tails stick outward from the cell and inward to the cell. And also in DNA and RNA, the pi stack of the nucleic acids line up in what's called the pi stack. So we have these aromatic confluences in pretty much all biomolecular structures, which could mediate quantum effects. Now, in the 60s and 70s, this gentleman, Herbert Froelich, who was a superconductivity physicist who turned to biology, proposed a feasible quantum state in living systems at ambient temperature. He said that coupled dipoles, like I just showed you between the aromatic rings, in nonpolar pi resonance regions that are in geometric array, so they're in lattices equally spaced, would couple and oscillate coherently in terahertz, 10 to the 12th hertz, gigahertz, 10 to the 9th hertz, and or megahertz, 10 to the 6th hertz in geometrical lattices. And the frequency would depend on the mass. So smaller things will oscillate faster, and as you get bigger and bigger, like more of a microtubule, it's going to get slower and slower. And we'll see more about that in a second. So what we need then is a geometric array of nonpolar pi resonance groups, and the microtubules shown here are very good candidates. If we look at them in the context of backing away a little bit, we can see that they are inside neurons or inside all cells. In neurons on the left, they are interrupted and of mixed polarity on the opposites, on the receiving side of the synapse. And on the right, you can see that some automata, cellular automata models we did back in the 80s and 90s showing propagating signals and patterns in microtubules and also the lattice patterns. So I thought that microtubules were a good candidate for Froelich coherence. Froelich agreed when I met him in 1986, and I was invited to write a chapter in his book. But then it fell to Anirban Bandyopadhyay to actually demonstrate this in microtubules. And he did it in this picture actually this is over 10 years of work over multiple papers, which were summarized in 2020 or in 2014. On the top, we have a couple of neurons, and the probes are nanoprobes. So there are many, many microtubules in those neurons. Then in B, we have one microtubule that's contacted by 10 nanoprobes. And then in the lower one, we have 2 rows of tubulin, even smaller, with 4 nanoprobes. And the experiment was pretty much the same at all 3 scales, where they used 2 electrodes to stimulate and 2 to record. And if you just put a voltage across a microtubule, it's a good insulator. But if you apply an alternating voltage or an alternating current at given frequencies and sweep the frequencies, you will find, or Anirban found, certain frequencies that the microtubule becomes highly conductive, possibly superconductive, but we called it ballistic conductance because there's a classical interface between the probe and the surface. And he found these patterns that are triplets of triplets. If you look on the right, you'll see each peak has 3 peaks, and each of those peaks has 3 peaks. So a triplet of triplets. And this repeated starting at the top in hertz, kilohertz, megahertz, and then at the mid-range, kilohertz, megahertz, and gigahertz, and then at the smallest range, megahertz, gigahertz, and hertz. And in all cases, there are triplets every 3 orders of magnitude. So these triplets of triplets may be very important. And we're also seeing something like that in samples from extraterrestrial molecules, that my work with Dante Loretta, who's here, that we'll be talking about later in the meeting. So some people say, well, okay, why not actin? Actin and tubulin are the 2 most prevalent proteins in the brain, each about 15%. And if we look at a microtubule and map just the tryptophans, which is the most responsive aromatic ring, you can see on the right how they're spaced in 3 protofilaments. And if you curve them, then they would get closer on the internal side. And the distance, at least going up the protofilaments, is within the Forster distance, so it's going to be a quantum interaction. If we look at actin, there are tryptophans, but they're further apart and above the Forster limits. So this is less likely to have quantum effects. So the microtubules do. So let's zoom in on one tubulin, and we see that there are 3 aromatic amino acids, tryptophan, phenylalanine, and that should be tyrosine at the bottom. And there are 86 of them in each tubulin. That's a huge amount for a protein. And the red spheres are where anesthetics bind. And, excuse me. So we found pathways through these aromatic rings that would extend into the lattice. So for example, this one going from southwest to northeast very roughly, and we just simplified it with a few rings and found identified these pathways as quantum channels in tubulin. And this was the basis for the qubit and the orcho-R theory, where we have oscillating orientations of the dipole pathways and a superposition of both pathways, both directions, that travel in this helical arrangement, helix around the microtubule, extending the length of the microtubule. So in a axon in a sciatic nerve, it can be a meter or longer, and a giraffe even longer. And in dendrites, it's shorter because they're interrupted. But these can be mesoscopic length quantum effects. That are buried and sequestered inside the protein, so they're not in contact with the water. And these are called Froelich giant dipole oscillations. So neurons have a lot of microtubules, as you can see here in this cover of Neuroscience. And here they're continuous, so they're probably it's probably from an axon. And the quantum aromatic channels lie within mesoscopic microtubules, which pervade neurons in the brain, and we call this the quantum underground. We published a paper. Travis was first author on the quantum underground, and basically saying that if you consider the quantum channels, 13 quantum channels and the 13 protofilaments of each of these microtubules, there's a lot of quantum channel in there, even if they're tiny, because they have to be tiny, so they're buried deep within, but there's a lot of them. And when you think about the entire brain with all the neurons and all the glia, I mean, glia have microtubules also, there's a lot of quantum underground going on, just in the microtubules, plus in the other structures I mentioned earlier. So I think that the region of the nonpolar quantum underground is a region of adiabatic decoherence-free subspaces oscillating coherently within microtubules and other structures separated from the warm, wet, and noisy brain. And finally, some new work from, well, it's not that new actually, but I hadn't appreciated it till recently, showing entanglement between microtubules using these, finding these optical vortices. And Anirban's here, I think, so he can maybe explain this if we have some questions. Doing Bell's inequality measurement on a pair of microtubules where you stimulate with UV, I think, and you get these projected optical vortices. And then at the bottom, they determine that angular momentum between the two microtubules, which were once together, then separated, were entangled. This is in Saxena et al. From 2020, I think. And so this shows at room temperature entanglement between microtubules. And if we have entanglement between microtubules in a neuron and between neurons, then in principle the whole brain could be entangled. And I think the brain needs this. I think we need this to explain things like brain-wide gamma synchrony, which is, I think, impossible to explain by classical mechanisms and nerve propagation and even ephaptic transmission, because it kind of peters out after a while. So I think the brain needs to be entangled for it to work properly, and this is the first inkling that entanglement does occur, and I hope that there will be more. So let me conclude by saying that, in my opinion at least, and Schrodinger's, life is quantum in nature. Its essence is due to collective oscillations in adiabatic decoherence-free subspaces, what we call the Meyer-Overton quantum underground. Meyer-Overton because they discovered the site of the medium of anesthetic action. The quantum underground includes sequestered aromatic rings and other nonpolar biomolecules whose activities can entangle into unitary states. And consciousness in the brain is likely to occur in a scale invariant hierarchy in mixed polarity microtubules in cortical layer 5 pyramidal dendrites and soma. And the brain is more like a quantum orchestra than a computer, certainly more so than a classical computer. and different from a regular quantum computer. It's, there is a scale invariant hierarchy. I didn't show this slide, where we have triplets of triplets from hertz, kilohertz, megahertz, gigahertz, terahertz, and petahertz, every 3 orders of magnitude. And within this, consciousness can occur and entanglement can occur, and that's how I think this is going to prove to be necessary for brain function. Thank you very much. Yeah.
Hello. Thank you so much for for the great talk. I have been always interested in Alzheimer and the tau protein. So, do you see any possibility of the interaction with microtubule and microtubule associated proteins map like tau or others?
Are you talking about Alzheimer's? Is that what
Yeah.
you said for? Okay. Yeah. Yes, definitely. Tau is a microtubule-associated protein. It actually functions as a traffic signal on the microtubule. So when the motor proteins are moving along to deliver their cargo to this particular synapse, the tau tells them, get off here, because that particular synapse needs more enzyme or whatever it is they're carrying. So I think Alzheimer's is a disease of microtubules. And this was pointed out in a paper in 1989 by Matsuyama and Jarvik of UCLA in Proceedings National Academy of Science. Hypothesis, Alzheimer's is a disease of microtubules. And they nailed it. I think they were exactly right. And I thought they were right. And, but unfortunately, big medicine, big pharma has gone into hammering away at the amyloid plaques, which don't work. I mean, they're toxic, they're ineffective, they're expensive, and they just don't work. And I've been trying to do a study for years. We actually did a pilot study. Transcranial ultrasound is megahertz, and one of the vibrational frequencies is megahertz. And if you put low intensity, some people tried at high intensity, and some people tried at high intensity to zap the amyloid plaques. That's a mistake, I think. If low intensity resonates the microtubules and causes them to regrow. And that's what needs to be done. And we're going to try to do that study.
Can I ask one more question? Can I ask one more question? So during listening to your lecture, I thought if there is a tubulin mutation, if that doesn't have a tryptophan ring, but still serves as a skeleton, cytoskeleton, then if that animal or organism is unconscious, it'll directly prove your theory. Do you have?
I mean, a zombie? It would be a zombie, but
I don't know. Yeah.
Yeah, I don't know if there are any viable mutations without tryptophan. There are some mutations, Chediak-Higashi disease that causes problems with cilia, but I don't know of any mutations that take away the tryptophan or the aromatics, and it's still viable.
Thank you.
You're welcome. Some over there.
Hi, very interesting. I'm not sure
Thank
I
you.
understand it all, but what I'm wondering is, so the biological evidence that consciousness seems to be a quantum phenomenon comes from these studies with anesthetics, right? So,
Well, that's evidence. That's evidence. Yeah.
So I guess what I don't understand is why these specific anesthetics like xenon disrupt, disrupt consciousness, and I guess they also disrupt quantum phenomenon, we're assuming. I guess I didn't quite catch that
It's
part.
not just xenon, it's all the anesthetic. That's a very good point. So I'm reminded 30 years ago or so, I asked, talking to Christoph Koch, who'll be here, about a couple things, and I said, we really should understand anesthesia, and that'll help us understand consciousness. And he said, no, because anesthesia acts very non-specifically. It binds all over the body. It binds to everything. So how can we possibly, we need something specific that gets at the specific site or target of anesthetic. And I didn't have a good answer for that, because it's true that at 1 Mac, the patient has a huge amount of anesthesia in his or her body. And I often tell the residents when I was teaching them, there's more anesthetic in the guy's butt than his brain, but it's probably working in the brain as a joke. But it's all over the place, and yet all it affects is consciousness. And I pondered that for many years, and a couple months ago, I think I figured it out. And the answer is that anesthetics disrupt any organized quantum effect or randomize any quantum interaction. But the only organized quantum activity is for consciousness. Everything else is already randomized. So it can randomize the quantum stuff in the butt or anywhere in the neuron that's not directly related to consciousness. It's not going to matter because they're already randomized. So consciousness involves and requires orchestrated or organized collective quantum interactions, like a Bose-Einstein condensate. If you mess up one part of it, the whole thing is shot. So I think that's what happens.
Okay, I think I understand now. So is that the only sort of convincing biological evidence that consciousness is a quantum phenomenon?
Is that the only? No, I think there's others that probably shouldn't go into now, but brain-wide gamma synchrony. I don't think you can explain that. Neuroscientists might argue with me, but I don't think they can. Real-time conscious action, because if you say something and my brain is processing, I answer before the processing has occurred. So the party line is that it's a zombie. My unconscious autopilot is answering, and I have this false illusion after the fact that I answered consciously. That's what the party line in neuroscience is, and that makes, that means no free will. There's a lot of problems with that, but that's what they say. Easy to get around that in quantum because you have this backward time effect that Benjamin Libet showed in the 70s and that Roger Penrose has been working on lately. And there's some others. The unity of binding, how everything is bound together. You process space, shape, color, motion, meaning, different parts of the cortex, different places, different times, and yet you see one object with one set of characteristics, not the characteristics separately. So that requires spatiotemporal binding, which you need entanglement for.
Excellent, thank you.
Hi. My question relates actually a little bit to the first couple of questions about tubulinopathies. Moving on to iatrogenic tubulinopathies, there are various drugs that, sorry, is my accent?
No, it's my hearing too, but go ahead.
There are various drugs that disturb microtubule function or structure in various ways, Vinca drugs and Taxols and colchicine and things like this.
Yeah.
And I know that many of them don't cross the blood-brain barrier,
right.
but particularly the oncological drugs, there are new techniques now like ultrasound to perturb the blood-brain barrier and improve CNS delivery.
That's medium-dose ultrasound to open the blood-brain barrier, which is one way to do it. If you want a drug, if you've got a drug you want to get across, you do that to get it in. I don't think you need to do that. I think low intensity will affect the microtubule directly. But if you're asking about these, when we first proposed this, we were criticized by Pat Churchland, the famous philosopher, who said, well, people take colchicine for gout, which depolymerizes microtubules, and they don't lose consciousness. And I pointed out that colchicine only affects microtubules that are actively assembling and disassembling. In neurons, they're quite stable. It doesn't cross the blood-brain barrier. And in a study where they injected colchicine into a rat brain, it just wiped out the rat. So, now Taxol, by the way, which stabilizes microtubules, there's a clinical study in Eckenhoff's lab of patients' anesthetic requirements, and patients on Taxol, mostly women for breast cancer, unfortunately, required more anesthesia than everybody else. Because I think Jack Jasinski and Travis said it was maybe blocking access to the nonpolar site, or anyway, it stabilized to the point that it kind of antagonized the action of anesthetics. So you had to give more.
That's interesting. I wonder if there's room then for its use in neurodegenerative. I wonder if there's use then for Taxols in neurodegenerative diseases like Alzheimer's, if there is a tubulin-related dysfunction.
I just retired from anesthesia, but I was also always very, very careful about elderly patients because of the risk of precipitating or exacerbating Alzheimer's. And so what we can do is, first of all, keep them warm. If they get a little cold during anesthesia, that's not good for their microtubule stability. And avoid too many repetitive anesthetics. But older patients and babies who had serial anesthetics seem to be prone to postoperative cognitive dysfunction, which might be a kind of an Alzheimer's picture.
Thank you.
Are we okay for time, or? Huh? Okay. So should we stop now? Maybe one more. Okay.
Thanks, Stuart. When you say consciousness, do you mean sentience, or do you mean the opposite of unconscious?
You know, I still don't get the difference, and I between sentience and awareness and consciousness, and people say, well, define consciousness, and I tell a story about a conference where they were going to define it on the first day and figure it out the rest of the week. At the end of the week, they're still arguing about it. I know people make a distinction. I'm not sure of the distinction. Everything you say is a word. You know, awareness, experience, phenomenal experience, qualia, sentience, and they're all words. You know what it is. I know what it is, and I define it as what goes away with anesthesia and comes back when you wake up. I mean, because John Searle said it's what goes away in a dreamless sleep and comes back when you wake up, and I just adapted that to anesthesia.
Okay. Thank you.
Thank you. Thank you all.
Okay, great. So next up, we have Travis Craddock. Dr. Craddock is an associate professor at the Departments of Psychology and Neuroscience, Computer Science, and Clinical Immunology at Nova Southeastern University in Fort Lauderdale, Florida. He serves as the director of the Clinical Systems Biology Group at NSU's Institute for Neuroimmune Medicine, where he applies computational systems biology and biophysics methods towards the purpose of identifying novel treatments for complex chronic illness involving neuroinflammation. Dr. Craddock received his PhD in the field of biophysics at the University of Alberta, where his graduate research activities focused on subneural biomolecule information processing and nanoscale neuroscience descriptions of memory, consciousness, and cognitive dysfunction in neurodegenerative disorders. His molecular regulation of chronic illness resulting from exposure to neurotoxins such as anesthesia and nerve agents or viral infection in order to improve diagnosis and putative treatment strategies. His work is primarily funded by the U.S. Department of Defense and National Institutes of Health. So please let's all welcome Dr. Travis Craddock.
Hello, everyone. Can everyone hear me okay? The thumbs up? I'm assuming everyone can hear me. Yeah. Okay. Let me share my screen here. Okay, so first I'd like to thank the conference organizers for inviting me. I did intend to be there, but I had some pressing matters come up here where I wasn't able to travel. Thanks to Stuart again for the kind words in the earlier talk. I'm going to be piggybacking a lot off of what he said and kind of talking about the opposite side of some of the things. He was talking about anesthesia, which takes away consciousness, and I'm going to be talking more about psychedelics, which we consider to expand consciousness in a certain way. As you've heard, I'm from Nova Southeastern University in Fort Lauderdale, Florida. I'm at the Institute for Neuroimmune Medicine, where our prime goal is to find new diagnostics and treatments for neuroinflammatory illnesses. Primarily, that's Gulf War illness, chronic fatigue syndrome, and long COVID, but we've also branched out into things like Alzheimer's disease and Parkinson's disease. I'd like to thank my team, Dr. David Casada and Will Freeman, who form the backbone of our team, and then my students, Isadora, Caleb, and Tatum. And a lot of the work that we're going to be discussing today is done by those three students in my group. And then the funding provided for this work is mostly from institutional grants from NSU, Army Research Office grant, as well as a grant from the CDC. And all of the computation I'm going to be talking about was performed on the University of Miami high performance computing platform, Pegasus, down at the Center for Computational Science. Okay, so we heard from Stuart about microtubules, just a little bit more detail on what they are. These are hollow cylinders of the protein tubulin that form elaborate networks in every cell, all eukaryotic cells. In neurons, they form a specific architecture, which you see there on the left, where they bundle together in parallel or anti-parallel formations to give the rigidity and structure to dendrites and axons. Whereas on the right, those are other types of cells, so kidney cells, skin cells, glia, and this is where microtubules more form an aster-type complex radiating out from the nucleus. On the left, in neurons, they're mostly stable, specifically in kind of forming the core of the axon and the dendrites. But when you get into the axonal growth cone and into the dendritic spines, the microtubules can become quite dynamic and move around a lot, especially during learning and processes like that. Whereas in other cell types, microtubules are quite dynamic overall, changing shape and size depending on what they're responding to in the environment. Beyond maintaining the structure of the cell, they're also responsible for performing the train tracks, which carry intracellular cargo throughout the cell. And then it's been theorized that they form a role in signal transduction throughout the cell. But this is, again, just a theory at this point, although more and more evidence is pointing in that direction. Zooming in on the structure, there's the protein tubulin on the left. It's a heterodimer made up of two individual dimers, alpha and beta tubulin, which are never found alone within the cell. Once they're made, they form into these dimers. Then these dimers stick end to end on the far right to form these strands called protofilaments, and then these protofilaments stick side to side together, kind of offset, so that when they wrap around, they form a helical structure that winds up in a left-handed helix. The scale, they're about 25 nanometers in diameter, and they can range in length from micrometers to millimeters, depending on where they are in the cell. Now, because of the size of these protein structures, they're effectively a nano device. So it was one of the goals in nanotechnology or bionanotechnology is to mimic the properties of microtubules to try and provide either sensors within cells or a way to manipulate processes within cells by manipulating the cytoskeleton. Now, the closest thing in bionanotechnology that we have to this is a multi-walled carbon nanotube. And depending on how many layers of graphene wrapping around a central core, you can get diameters that are very comparable to a microtubule of being about 15 to 25 nanometers. And then depending on the length of the carbon nanotube, it can range around the same length of a microtubule itself. It's been shown that multi-walled carbon nanotubes can enhance microtubule assembly and stabilization depending on the state of the cell. It's also been shown that these multi-walled carbon nanotubes can inhibit the formation of microtubules, so they can either enhance stabilization or disrupt stabilization depending on how they're used. Now that's kind of using nanotechnology to mimic biology, but the kind of approach that we've kind of taken is trying to look at the tools of nanotechnology to study microtubules and look at what is it that goes on in carbon nanotubes that might be analogous that could go on in microtubules. So looking at them as optoelectronic devices or potentially being quantum information devices. And we've done a lot of modeling throughout the years to look at particularly these aromatic amino acids in microtubules and the potential for exciton transfer, which Stuart alluded to earlier, superradiance, which I'm hoping Phil Kurian will be discussing later, and some phaneresonant properties that we seem to have discovered with Robert Alfano at City College of New York. Now, Stuart already kind of showed this picture, so aromatic amino acids can effectively act as a chromophore. What that means is that they are light-sensitive molecules. So when they're excited with a photon of light, they get pushed into an excited state, And then after a period of time, they relax and release that energy back into a system as a photon of lower energy. And tryptophan just happens to be the most photoactive aromatic amino acid in biology, but there are other ones such as phenylalanine and tyrosine. Other ones that aren't a primary amino acid, but things like flavin, porphyrin rings, which you may hear from other individuals giving talks today. Chlorophyll is another example of a light-sensitive molecule that harvests energy in photosynthesis. We're applying this same sort of methodology from photosynthesis to understand these aromatic chromophores in microtubules. This is kind of what it looks like in a tubulin molecule. So on the left, you have tubulin with the tryptophan molecules highlighted in blue. And then if you strip away the rest of the protein structure and just look at the tryptophans, that's shown there on the right. And those are spacings in angstroms. So the spacing of these tryptophan chromophores is relatively comparable to the spacing of chlorophylls in a photosynthetic light harvesting antenna, and the energetics are fairly similar as well. And that gave us the impetus to try and study these using the formalism of light harvesting in photosynthetic units. We did those studies back in 2014 and later in 2017 with Philip Kurian. We simulated the propagation of an excitation, a single ultraviolet photon, exciting this network of amino acids, and then looked to see how far would that excitation travel along the surface of a microtubule. In this case, it was along the direction of a single protofilament. Because there's many free parameters and unknowns, we modeled various different situations. And depending on how stringent or how liberal we were being with those factors, that excitation could travel anywhere from within a single dimer up to traveling up to 12 tubulin dimers. One thing that was shown here, though, is that there's a preferential direction. You can see kind of everything is kind of shifted to the right there. And that means that excitation does prefer to go in one direction from the alpha tubulin monomer towards the beta tubulin down that direction. And that seems to give some sort of directionality of where the energy would like to travel. Stuart had mentioned this as well. This is work by Arat Kalra when he was doing a postdoc at Greg Scholl's lab. And what he did was he labeled tubulin monomers with a reporter protein called AMCA. And after varying the different concentrations of that, he was able to assess differences in the tryptophan emission lifetime. So he could measure the differences, how long it would take that excitation to travel from an initial tryptophan towards one of these AMCA reporting molecules, and look at from that time that it would take and the concentration of AMCA, he could figure out how long that excitation would travel along the length of a microtubule. Now, according to standard Forster theory, you heard Stuart talk about that. If that transfer was to take place between tryptophan and tyrosine along the surface of a microtubule, it would travel about 1.54 nanometers. But what they actually measured was diffusion length of about 6.6 nanometers, which is quite long compared to standard Forster theory. Why that is, it's not currently known. We're trying to look into that. So that's kind of this situation here, regular microtubules. And then if you add anesthetics you can see that distance is suppressed from about 6.5 nanometers down to below 6 nanometers. So it truncates the length of distance that excitation travels. And so this is in line with that theory that Stuart was talking about. Rod Eckenhoff at UPenn coined the term quantum mobility theory of anesthesia to describe Stuart's ideas. And that is that with inside microtubules, you have patches of tubulin, and inside those tubulin, you have these aromatic amino acids where anesthetics can then get in between them. And in normal situations, you have oscillations among these aromatic amino acids. And then when you throw in an anesthetic, because of the ability to form transient dipole moments, it can disrupt that oscillation and suppress the oscillation, these coherent quantum oscillations that you heard Stuart talking about. So that's the overall theory of the quantum mobility theory of anesthesia, and these kind of results, are in line with that idea. Again, in 2017, we decided to test that more concretely, theoretically. This is, again, work that I did with Philip Kurian, Jack Tazinski, and Stuart. And what we did is we did a computational experiment where we computationally docked anesthetic molecules to tubulin and then calculated how it would affect the coherent oscillation of all those aromatic amino acids within a tubulin molecule. And what we found is that the addition of an anesthetic adds an additional frequency mode. So tubulin alone, because there's 86 aromatic amino acids, there's many different ways that those aromatics can oscillate. And depending on how they oscillate, they oscillate at different frequencies. And those different frequencies are shown here between about 450 or 480 terahertz all the way up to about 700 terahertz. And those are the natural frequency modes available to the tubulin dimer. But when you add in an anesthetic, you add in an additional mode, and depending on the potency of that anesthetic and some of its polarizability, value, it will affect the frequency mode that it's oscillating at. Now, in blue here, we're showing anesthetics. And in pink, these are molecules that are predicted by the Meyer-Overton correlation to be anesthetics, but they are not. So these are called non-anesthetics. They get into the brain, they follow the Meyer-Overton curve, but they do not produce anesthesia. And we can see here that over to the right of this kind of 600 mark is where you get these anesthetic molecules, whereas to the left is where you get these non-anesthetic molecules. Furthermore, we looked at how each of these individual anesthetics affected those normal modes of tubulin. And what we found is that all of the anesthetics shift the normal modes of tubulin down by a few gigahertz, so 20 up to 100 and something gigahertz, depending on the anesthetic looked at. And the prime shifting always occurred near this 614 terahertz mark, or approximately 610 terahertz. However, the non-anesthetics, this F6, does not shift at that 610 terahertz, and this other non-anesthetic TFMB actually shifts it up at that same frequency range. And so if you plot those shifts, you get this, what we're calling kind of like a new Meyer-Overton curve, which is based on these shifts in these coherent oscillations, where you correctly predict the the potency of individual volatile anesthetics, but you also predict the potency of non-anesthetics, which is not readily predictable by just the Meyer-Overton curve. We haven't tested this in other proteins. We have not looked at this yet in terms of membrane proteins and other receptor proteins, but it is something we do want to look at. Now, what I wanted to get to you guys about is talking about psychedelics. Obviously, anesthesia shuts consciousness off. Anesthesia shuts consciousness off, and psychedelics change and alter your consciousness in various different ways depending on the compounds that you're looking at. And there are three main classes of classic psychedelics. These are tryptamines, lysergamides, and phenethylamines. And my lab is primarily looking at phenethylamine psychedelics. There's many different natural phenethylamines, and synthetic phenethylamines are kind of on the rise as new designer drugs. And it's not really sure how dangerous these things are. And so we're kind of looking at their effects on neuron viability, as well as the direct effect on the cytoskeleton. And they can have various effects. Dopamine and norepinephrine are phenethylamines that are naturally occurring in your body. They can have non-psychoactive effects like albuterol, which is an anti-asthma medication. They can be psychostimulants like methamphetamine, or they can have psychedelic effects like mescaline or MDMA. So back in the 90s, Van Wolkerum put out a hypothesis paper, and he looked at the structure of mescaline, shown here on the right, and he compared it to colchicine. And you can see here that you have that same benzene type structure with this trimethyl methylation on it, and this the amine backbone, which is shown here on colchicine. And the idea is that these psychedelics, at least the mescaline phenethylamine based psychedelics, are structurally similar to colchicine, and therefore they may act on the cytoskeleton directly. And there's some evidence for that in the past of mescaline binding directly to tubulin to alter axonal transport and inhibit microtubule formation in the mitotic spindle. 2-phenethylamine has been shown conversely to stabilize microtubules, so acting in opposite ways. One is inhibiting, one is stabilizing, but it does suggest a biochemical basis for direct interaction of these phenethylamines with tubulin and microtubules. So earlier last year, we published a molecular analysis where we did computational docking of phenethylamines to the colchicine binding site. We picked the ones that are of about 110 phenethylamine compounds, and we picked the top 3 that were most likely to bind to tubulin and looked at them experimentally to see how they would alter microtubule polymerization dynamics. And we found 2 of them, 25B NBF and 25C NBF, looked to stabilize microtubule at increasing dosages, whereas this other compound, DMB MPP, looked to destabilize microtubules. And through microscopy, we actually saw that effect is that DMBP forms these clumps of tubulin, whereas these 25B and 25C NBF compounds form intact microtubules at higher density. So it does seem to be that these phenethylamines, at least these 3, can bind directly to microtubules and alter polymerization dynamics. These also readily cross the blood-brain barrier, and so could easily cross into brain cells and interact not only with intracellular targets like the serotonin receptor, but also with things like the microtubule cytoskeleton. Now, since then, we wanted to see how would this affect this kind of quantum mobility theory of, not of anesthesia, but the quantum mobility theory of psychedelia. And so we've done computational binding of these phenethylamines to tubulin to get their conformation and their orientation, and then performed quantum chemistry calculations to find out molecular properties like their polarizability, solubility, their excitation values, so how easily are they to be excited by UV photons, at what wavelength are they excited, and so forth. Docking these into the tubulin complex and using that photosynthetic mechanism that we described, that we've done work with Philip earlier, we found that the compound DMB MPP can alter the energy landscape of that excitation, and would seem to suggest that if you did the same experiment with DMPP in tubulin that Caldera did with anesthetics, you would see an altered excitation pathway. So it would either increase or decrease that diffusion length. At this point, we're not quite sure which direction it would go. However, things like N25B NBF and 25C NBF, they have very, very weak transition dipole moments, so we would not expect to see any sort of alteration in that exciton traversal experiment. However, if we look at the way in which it could affect overall collective excitation modes like we did with anesthesia, we find that 25B NBF, 25C NBF, and DMBP all produce novel excitation modes way lower than what the anesthetics showed, and lower than what the traditional non-anesthetics did, well lower than any of the modes within tubulin. And so we're not quite sure what this means, but if anesthesia increases on the right, and then in the middle here you kind of get non-anesthesia, maybe down here on the left, this is why you might get something like enhanced psychedelic experience. It's not to say that this is any sort of slam dunk agreement with confirming this theory, but it's not inconsistent with quantum mobility theory at this point. And obviously, this is again theory in nature and something that we need to explore a little bit further and do more detailed analysis with multiple phenethylamines. But it is something that we find interesting and we think is worth exploring further. Here are the references that are cited throughout my presentation. And then I'd also like to announce that I'm currently at Nova Southeastern University, but starting May 1st, I'm going to be moving to the University of Waterloo in Ontario, Canada. I'm going to be joining up with the Department of Biology, where I've accepted a position as a Canada Research Chair. So I'm going to be focusing more on these quantum mechanisms within brain function, looking at the molecular properties of neurons, nano-neuroscience, with a prime goal of still looking at neuroinflammatory disorders, including Alzheimer's and Parkinson's disease. So if anyone's out there and they're looking for graduate student positions or postdoc positions, please reach out. And finally, I'll leave it up if we have any questions.
Okay, great. Thank you so much, Travis. So we have time for some questions. First question over here on the left.
Hello, I wish I knew which place to aim at, since I'm sort of talking to an anonymous something. My name's Bill Sofsky. I was delighted to learn of your work last November because the laws of representation in brains have predicted the existence of microtubules, because the brain needs some kind of lattice capable of representing three-dimensional reality at the resolution we do. When you're talking about teravoxels in nanoseconds, you need quantum coherence and you need three-dimensionality. So what I'm wondering, can all of these resonances in microtubules, if you could arrange them any which way, could they form a lattice metamaterial equivalent to like a cloud chamber which moves, an imaging metamaterial which could sustain three-dimensional waves at multiple velocities? So you could have the wave traveling through material and then dial some resonances and slow it down the way they slow down the speed of light. Can you make such a thing for microtubules, maybe?
Short answer, I'm not sure. That's not really my area of expertise. I think, you may be able to conceive something like that. It would have to, correspond with the, physical parameters available to microtubules. So there's only given ranges that are available, at least with these aromatic oscillations that I was showing. There's a set range, roughly between 500 and 700 terahertz. There's the spatial scale of microtubules. So between individual tubulins, we're looking at 8 nanometer distances, ranging from 4 nanometers to 8 nanometer distances, depending on which way you're going. But then between microtubules themselves, they can be spaced apart by about 50 nanometers. So they're about 25 nanometers in length, and then there's twice that, between each one. And these are now then connected with structural proteins like tau or MAP2, MAP1, and those will give different distances between the two. So there may be a way to figure that out. I'm not as familiar with these 3D representations of was it visual experience, or what was it we were trying to?
To be continued. Thank you for enough detail. I know what to ask later on.
Okay, perfect.
Thank you. Yes.
Hi, Travis.
Hello.
I've heard you and Stuart talking about, I guess, the active ingredient in psychedelics being these benzene rings, and just how they donate electron resonance to quantum dipole oscillations. And I never hear anyone ever talking about cannabis. It has that benzene ring, and people who smoke cannabis rarely have mystical experiences where they go into the light and the whole 9 yards. So do you see any fruit to bear?
So we have actually, Stuart had asked me a while ago to look at THC binding to tubulin, and we've done it. And we were looking at the difference between delta 8 and delta 9 THC. I'm not sure if you're familiar with that, but one is is what you get in regular, homegrown, not homegrown, but marijuana or cannabis that you get from the plant. Whereas Delta 8, I believe, is more of a synthetic version of THC, and one is more potent than the other. And the only thing that's different is one of those double bonds with one of the ring structures is, instead of being to the left, it's to the right of a given bond. And from standard chemistry, basic chemistry, that shouldn't seem to make a difference. However, it does, and we're trying to look at why that is. In regards to why you would not get a psychedelic experience with THC, I've heard other people who have anecdotally spoken about psychedelic experience. However, it's at very large doses of THC. So there may be a threshold that needs to be crossed in terms of dosage. And again, you'd have to look at how much it would affect any sort of oscillations within these tubulin proteins. So it might just be a matter of strength as opposed to whether it does or doesn't occur. That being said, again, this is all theoretical. We're kind of playing around with these ideas and seeing, what stands and what holds and what can't occur. So stay tuned for some results on THC coming out.
Thank you.
Yes.
Hi. Just in simplistic terms, I mean, what you seem to be saying is that anesthetics dampens consciousness and the psychedelics increases consciousness. Is that what you're saying?
Yeah, that's the basic tenet of that quantum mobility theory of anesthesia.
Okay, so what effect does increasing consciousness have? Like, what do you study? What do you show? The psychedelic effects then of increased consciousness. What evidence is
What
there
do
of
I
that?
show?
Yeah, what does it show? Like,
Oh,
what does that
so
mean?
I don't look phenomenologically at anything. So all I'm looking at is kind of the physics of the situation here. So the theory goes is that that these oscillations in tubulin are related to conscious experience. And if you dampen them, that you take away conscious experience, so you no longer have these oscillations occurring, or they're occurring at a lower rate, which would then somehow affect timing or some sort of resonance effects or entanglement within the brain. That mechanism is not worked out. It's kind of hand wavy, that's why I'm doing this. The other way would be that if these psychedelics would enhance these oscillations, again, it would work the other way. It would increase the coherence, or it would increase the entanglement, and would allow for a greater processing of information. Again, very, very hand wavy. I'm looking at this from a kind of a nuts and bolts perspective. Sure. Is that there's the overarching theory that Stuart has put forth, and looking at what we know about microtubules biophysically, what we know about these anesthetics and psychedelics physically and quantum, based on quantum chemistry. Does that theory hold weight? And all I'm saying is that it's not unfeasible.
Right. So, but when is there any measurement of the increase of the processing of information?
Not that I know of. It's just basically. Are you talking about kind of at that lower scale, kind of where I'm studying molecular?
Yeah,
or are you talking
when the psychedelics are affecting the microtubules, is there any measurement then of increased, processing power of the brain?
So no, I mean not as far as I know. So I know on this molecular scale you can do experiments to measure whether, for example, that the cholera work, you could throw in a psychedelic, measure to see how far that excitation travels, and if it's longer than what is normally available, then you could say that's consistent with quantum mobility theory. Whether that enhances computational ability of the brain, I don't know how to do that. At the far end, you can always get phenomenological experience. So someone takes a psychedelic and then reports, greater awareness, greater experiences, expanded ideas about or visual hallucinations and so forth. That's the far end. That chunk in between, going from this very, very molecular scale to the actual experience, that is a big chasm. That's something that I haven't worked out, that I don't know if anybody's worked out at this point.
Thank you.
All right, thank you so much, Travis. I think we're
Thank
going
you.
to move on to the next speaker. I'd like to welcome Dr. Albert Siryaporn. Dr. Siryaporn received his PhD in physics and astronomy from the University of Pennsylvania, exploring how bacteria limit crosstalk between different signal transduction systems. He was a postdoctoral fellow at Princeton University in the Department of Molecular Biology, where he investigated how bacteria interpret and respond to mechanical stimuli. He received the NIH NRSA Postdoctoral Fellowship and NIH K-22 Faculty Transition Award. And he joined the Department of Physics and Astronomy and Department of Molecular Biology and Biochemistry at the University of California at Irvine in 2016, and has received major research grants from the NIH. His lab uses approaches from systems biology, biological physics, and molecular biology to investigate the dynamics of life in bacteria.
Great. Thank you very much for the organizers for inviting me. I'm going to tell you today about exploring different quantum sensors of living systems. And we're going to start with sensors that a living organism has that can detect kind of quantum level phenomena. And then we'll go into some of the synthetic sensors that we have built in order to detect quantum phenomena. And the kind of the level of this talk was really to give an introduction and exploration of kind of quantum biology. And so we'll dive right into it. Okay, and I did want to point out, so my appointment is in Department of Physics and Astronomy. I also have an appointment in molecular biology and biochemistry. And so this is, my work is inherently interdisciplinary between physics and biology. Okay, so we'll start with quantum biology and trying to motivate for why do we need quantum mechanics to understand biological systems. And so living systems have been described in the past using classical mechanics. We think of things as such as forces, we have fluids, and we have electrostatics. And this has served us very well for a long time. And so if you think about kind of very basic functions, such as something that is involved in motility. Okay, so bacteria have these structures called flagella that allow them to move. We have something similar in our bodies called cilia, which allow us to move fluids. And we've dived really deeply into how these mechanisms work. Okay? And so if you look at this, it actually looks a lot like how we would design an electrical motor. And what's happened over the course of time, so this is an image that was taken in 2006. If you look at what we know now about the structure of the same flagella, it's at much higher resolution. Okay? And so the resolution goes now to the subatomic level. And so what we need is when we reach this length scale, what we need is a different type of physics to understand what's going on. Okay? And so that's where we need quantum mechanics. We need to understand what's happening at the subatomic level. So I'm going to talk about two things today. So what biological mechanisms require consideration of quantum mechanics, and then how can we use quantum mechanics to aid as an investigative tool. So I'm just going to talk about 1 and 2 here. So exploring a quantum sensor from a living system. We're going to talk about how Magneto-reception and magnetosensitivity can work. And then we'll talk about quantum sensors that have been used to make new measurements. Okay, so my talk is going to kind of dive and really revolve around electrons. Okay, and we're going to talk about electrons as a central theme. And what you should know is that magnetism is very closely linked to moving electrons. That's what we would call that is electron dynamics. And I think one thing that's really important to understand during this talk is that electron states can be affected by magnetic fields. Okay? And so it's a quantum effect. So magnetic fields will actually alter the allowed energy levels of electrons. Okay? So knowing that, what we're going to do is we're going to look at how magnetic fields can alter different biological materials. Okay, we're going to need a little bit more introduction on electron and energy levels before we dive into this. Okay? And so what we have here is depicted are electrons. And electrons have this property called spins. You may have heard of electron spins. And so electrons can be paired, as they are here in this ground state. So you have two electrons. One is pointing spin up, one is pointing spin down. And what we have is when you put this in a magnetic field, the electron energy levels can change. So you have this introduction of a new energy level, and these electrons can take different states. This electron can go into a higher energy level, and the spins can also take a different state, where the spins point in the same direction. And so this, in this singlet excited state, this total spin here is 0, but in here, this triplet state is what we call they have unpaired electrons. Okay, so they're both pointing, there's a net spin in this system. And what happens here is that this can potentially change what a protein does. Okay, and so the singlet and triplet states could have different effects on chemical reactions. And so that's how we're going to explore the effect of magnetic fields on biological materials moving forward. Okay, so when would a living system be exposed to a magnetic field? Okay. And so the Earth has a magnetic field, and it's equal to about 0.5 Gauss or 50 microtesla. Just keep that range in mind as we explore this. There are a lot of artificial sources that we have created that have much larger magnetic fields, and so that's also something to consider. So we have two parts of this equation. Where do magnetic fields arise? Okay, so we have the Earth as a nice source for that, and we have other sources that are artificial. But then how can they affect a biological material? Okay, and so if they are going to affect a biological material, it should have unpaired electrons. Okay, so this is the triplet state that I was talking about. They have to potentially go into this triplet state. Okay, and so one area that this could arise where people have looked is looking at a ubiquitous molecule that's involved in metabolism. It's called flavin adenine dinucleotide, or FAD. Okay, so this is involved in many processes inside cells. It's so ubiquitous that if you take a look at an ordinary cell, in this case this is an epithelial cell, and you don't put any kind of dye in it, you don't modify the cell in any way, you just take a natural epithelial cell and you put it under the microscope, it's going to fluoresce. It's going to give off this light intensity that is at a different wavelength than what was used to excite it. And so what's really interesting here is that this FAD molecule can potentially go into this triplet state. And so a mechanism has been proposed that what happens is they form a radical pair. Okay? So the idea is that if you have FAD, you can excite it, and then you have electrons that are now potentially able to transition between the singlet and this triplet state. So the singlet state is over here, and the triplet state is over here. And I think that what's really important here is to understand that these are different chemical reactions, or these lead to different chemical transitions. So the singlet state can lead to a decay back down to the ground state at a different rate than a triplet state would. Okay, and so this is the basis of what we call the radical pair mechanism. And the magnetic field will affect kind of how many electrons will be in the singlet or triplet states. And so the takeaway here is that if there is a magnetic field, what you should take away from this is that it opens up another chemical reaction pathway, and that can lead to some altered biological signaling. Okay, and so we're looking at, this study is done by the Woodward Lab. Okay, and so this was done fairly recently. And the idea is, let's just take an ordinary cell, and what we'll do is we'll measure the fluorescence that comes out of it. And we're going to try to change the magnetic field that's near these cells. This is going to be millitesla, so it's going to be a little bit higher than Earth's magnetic field. But what they're going to do here is they're going to measure the fluorescence intensity as a function of time. Okay, and they're going to measure this as a function of how much of the magnetic field is around. And what you'll notice is that with time, this fluorescent signal decays. Okay, so what happens is basically the protein stops emitting, stops fluorescing. And but what you see is when you have a magnetic field around, it's going to oscillate around this decay. Okay, and if you zoom into that, you can look at these residuals. This is the residual fluorescence, and you're going to see that they're anti-correlated with the magnetic field. So the magnetic field goes up and down. It's oscillating here. and this magnetic field is going to change the fluorescence that's around in this. And this correlation, this anti-correlation, is pretty strong. Okay, and so this is kind of one very simple way of showing that a cell, a cellular metabolite, which is ubiquitous in humans, let's say, is altered by a magnetic field. Okay, so the fluorescence, the properties of this one molecule is already altered by a magnetic field. And so this raises the question, this is a ubiquitous molecule, where else could this molecule FAD be involved? Could this be involved in sensing quantum effects in some other way? Okay, and so we're going to turn now to a more complicated system, or let's say a more complex collection of cells. In this case, we're going to talk about this garden warbler, and we're going to talk about how birds could potentially sense magnetic fields. Okay, and so this has been an outstanding question for some time, how living organisms can actually detect a magnetic field. Okay, and so we know very well that birds migrate towards south for the winter, and they navigate using magnetoreception. And so what was proposed is that a similar mechanism involving FAD and a protein called cryptochrome, which is a light sensor, could be responsible for magnetoreception. Okay, and so this was proposed by Klaus Stolten in 1978. And so the idea is that the landscape is similar, that you have this molecule, cryptochrome and FAD together. The idea would be that light will activate this pair. Then you would have electrons that could potentially be unpaired. So the electrons could be in a singlet state, or with the magnetic field, they could be pushed into this triplet state. Each of these reactions is going to lead them down to a different path. The singlet path will lead to one chemical species, whereas the triplet is going to lead to a different chemical species. And so this was the proposal. And so since then, I think a lot has been done to show that cryptochrome has been really responsible for magnetoreception. Okay? And so cryptochromes are expressed in birds when they're performing magnetic orientation. Cryptochromes are found in the bird retinae, and so the idea is that it's involved in vision somehow. Light processing in the forebrain is required for magnetic orientation. And then weak magnetic fields, such as the ones that are on the order of the Earth's magnetic field, can disrupt the bird's magnetic orientation. Okay, so there's a lot of evidence that cryptochrome is involved in this bird magnetic orientation. Some further evidence is that the radical pair intermediates are observed from cryptochrome lyase activity. That's a requirement for this radical pair mechanism to work. And that the radical pair production reaction could respond to magnetic fields that are as weak as the ones that we find on Earth. Okay? And so those are all really strong evidence that cryptochrome is involved. So more recently, work by Peter Hoare's group showed that if you take this cryptochrome protein, okay, again, this is the light sensor, and then you put it in a magnetic field, what's going to happen is that it's going to change the spectroscopy, these kind of chemical properties of cryptochrome. And you could do this with the European garden warbler at different pHs. And what you see is that as the magnetic field increases, the magnetic effect, the amplitude of this magnetic effect is going to increase. It does, for a different pH, pH 8, the amplitude is even higher. And so these are maybe local effects that can tune how strong magnetic field sensing is. If you take the same protein, which happens to be present in a plant, they do see that there is a magnetic effect with higher magnetic field strengths, but it's not as strong as what one would see inside the garden warbler. Okay? And so this is also kind of consistent with the idea that in order for birds to magnetosense and navigate, they would need a stronger signal. Okay, so that's great. So this was a kind of really breakthrough discovery that you could take a magnetic field and you'd see these really well-defined chemical and biological effects. Okay, and so kind of the next step in trying to understand this is how we can take this and understand this from a chemical point of view, but how does this lead to something which is involved in sensing? Okay, and so sensing, as was alluded to before, there's a big gap between taking something that is a chemical mechanism to something that would lead to sensation within an organism. Okay, and so the idea here is that what has been proposed is that there are photoreceptors around, and inside these different photoreceptor domains, cryptochrome could be embedded and oriented within these domains. And that alignment is really important for understanding how you could tell if one is oriented with a magnetic field or not. Right? So these magnetic domains would have to stay in place. And so that's a great model. And what we're still missing is this gap between, okay, if I have a different chemical state, from either the singlet or triplet, how does that lead to a signal transduction system? Okay? And so when we talk about biochemistry, and we start talking about how proteins signal with each other, signal transduction is kind of how you get from sensation to transduction, or effect of some kind of protein cascade coming out. And that's the part that's missing. So we have a nice sensor here, potentially cryptochrome, but we don't understand how this could lead to a biological signal. Biological signals many times are done through phosphorylation, for example. This is where a phosphate gets exchanged and that changes the state of a protein. We don't understand how that would work with magnetoreception yet. And so that's something that my lab has really explored. And so we've looked at cryptochrome. This is the structure of cryptochrome. And what was brought to our attention is that the C-terminal domain, that is the kind of the end of this protein, is active. It's active in a way that it moves around. And so what we did was we constructed a garden warbler synthetic circuit. Okay, so we took the cryptochrome from garden warbler, and we attached it to another signaling system that we could get really fine sensitivity out. We describe it as the circuit as a signal integrator or accumulator, and the idea is that very small magnetic effects could be measured in this phosphorylation signal transduction system. So we created what's called a cryptochrome histidine kinase chimera. And histidine kinases are part of signal transduction systems which are really sensitive, really robust, really characterized. And if we saw any system, any kind of output from this magnetic system, we could potentially resolve it as a different type of fluorescence. Okay? So this would produce this cyan fluorescent protein. We've created chambers so that we can study this problem in bacteria. So we're studying an organismal effect inside bacteria, which is a very well-controlled environment. We don't have a lot of other signal transduction systems interfering with it. And then we have these magnetic chambers, which will finally control the magnetic effect. And so what we're doing here is, these are individual bacteria. We are measuring the output of these bacteria using the cyan fluorescence protein. And what we find is with this chimera, so this is the cryptochrome histidine kinase chimera, is that we can activate this with light. Okay, so there's some kind of a little bit light activation, and then but with a magnetic field around, the effect is much higher. Okay, and so what we're looking at is a biochemical potentially response from a magnetic field that is on the order of the Earth's magnetic field. So we're doing this at 50 microtesla, which is very small. So the preliminary experiments, and I do have to emphasize that these are very preliminary experiments. There's a lot more to do that we have to, in order to characterize how robust this response is. The preliminary experiments suggest that there is a magnetoreceptive response in this photoreceptor from the garden warbler. And what we're trying to work on really now is disentangling a light response from different magnetic field response. So how does it respond to different magnetic fields? How does the timing of this matter? All sorts of parts of this equation. Okay? And so what I wanted to leave you here with this first part of the talk is that cryptochrome and FAD are magnetosensitive. Okay? And that's really a large breakthrough when we're talking about biochemistry. And they demonstrate quantum sensing potentially by living systems. Okay. Second part of the talk I want to go over was using quantum sensors to make new measurements. Okay, and so what's really interesting about cryptochrome and FAD is that they are examples of living systems that can have already become sensitive to magnetic fields. So that's kind of one direct example of a quantum sensing from a living system. Can we use technology to, along the same lines, to detect quantum effects, quantum phenomena inside living organisms? And so one place to start, again, we have kind of two aspects that we have to think about. So where would you find unpaired electrons inside a living organism, and then how would you detect them? We need these unpaired electrons in order to have these magnetic effects. Okay, and so there are many places where there are formation of unpaired electrons. This happens during free radical production. So free radical production happens all the time inside living organisms. You have the example of UV light. Okay, UV light will create this reactive oxygen species. You have inflammation, metabolism, ionizing radiation, pollution, you name it. There's all sorts of sources that can cause unpaired electrons to arise. Okay, these produce these different reactive oxygen species, such as superoxide, peroxide, and so on and so forth. And so these can cause stress in the mitochondria, which is the powerhouse of our cells. They can also induce stress in other organisms. And so one of the examples that is given here is stress inside bacteria. Okay, so how can these species be detected? Now we move into the exciting field of kind of quantum materials. One of these quantum materials that has really emerged is nitrogen vacancy diamonds, these NV center diamonds. Okay, and again, remember that electron states are affected by magnetic fields. That's something to remember here. And potentially what we can see is that unpaired electrons that are in the system can be detected by this NV center diamond, this quantum material. This is conceptually similar to NMR, so we've heard of nuclear magnetic resonance, and so this technology has analogs to NMR, but is really kind of fine-tuned to detect this at a much smaller level. I'm going to give you two examples, and I'll finish, of how these NV center diamonds have been used. One is where NV centers, these diamonds, have been absorbed by cells. In this case, these are fluorescent nanodiamonds, and they're being absorbed by macrophages. Macrophages are immune cells that take up particles. In this case, they've taken up diamonds. And when these diamonds are targeted to the mitochondrion, which is the powerhouse of the cell, what one can start doing is now doing some quantum sensing to understand what's happening in these mitochondria. Okay? And so what the research is going to do is look at ROS production. They're going to cause ROS production by putting a toxin. This removes the ability of this mitochondrion to work. It's going to start producing all these reactive oxygen species, and potentially these electrons are going to be sensed by these diamonds. Okay? These unpaired electrons from ROS could affect NV diamonds. And I'm not going to go into too deep into the physics of what's going on, but there's something called relaxation time. So in NMR, you may have heard you pulse with a magnetic field, and then it decays through this relaxation, and there's something called T1 relaxation time. And what these authors showed was that if you pulse light into the system, you can measure these relaxation times. You can measure the quantum state within a living system. And so we see here that diamonds are being targeted appropriately to the mitochondria. And really what's interesting is now as you poison the powerhouse of the cell, as you poison the mitochondria, you're going to see these different shifts in this relaxation time, T1. Okay? So this is a quantum measurement. This is a very solid quantum measurement, and it's inside a living system. And it's inside, and what you see here is that when you poison the living system, you see this effect. It goes down. As you increase the poison, the effect will go down. And if you use a different poison, the effect will change as well. Okay, and so this is really supportive evidence that mitochondria toxins can alter this quantum state of, or be detected by this quantum sensor. Okay, so quantum ROS detected using fluorescent NV center diamonds. Okay, the last example I'll give you is that NV centers can be used to detect stress inside bacteria. So bacteria is what we mostly focus on in my lab. And one of the kind of emerging problems is that there's a resistance to antibiotics. This is called antibiotic resistance. And this has become a problem because the antibiotics that we have are becoming less and less effective. And so we're running out of antibiotics. And so what we like is to be able to understand how bacteria respond to antibiotics. And what this group has shown is that you can do that with these quantum sensors. Okay, so the idea here is that they're going to stress Staphylococcus aureus. You may have heard this bug in the context of clinical microbiology. The idea is that you go in, Staph aureus is kind of around, and when you go into the hospital, you can get an infection by an opportunistic pathogen. One of these is Staph aureus, so MRSA is kind of more commonly what it's known as. And what they've done here is they induce stress using vancomycin. And what they show is that with vancomycin stress, free radical production is increased, we know that, and that this free radical production can be sensed by these quantum sensors, these NV diamonds. And so you can see that as you increase the antibiotic concentration here, this T1 relaxation time will change in a antibiotic-dependent way. Okay, and so what I hope I've given you kind of introduced you to is that NV centers are quantum materials that can potentially detect different electron states within different organisms. They specifically measure the presence of unpaired electrons, such as ROS species, and they potentially have a broader detection applications. Okay, so there are many ways that NV centers could potentially be involved in detecting kind of quantum phenomena inside living organisms. And that's really where the state of the field is. And in the first part of the talk, I talked about magnetic field sensing, or magnetoreception. And the idea is that there's a lot of evidence that cryptochrome is magnetosensitive, and this could lead to the ability to guide organisms inside magnetic fields. The mechanism that was suggested, been proposed, is radical pair production. There's gaining evidence that this is the mechanism. And this leads to a triplet state where you have unpaired electrons. The mechanism of signal transduction remains to be determined. That is, how biochemically something can, how you take this quantum signal and translate it into a biochemical signal. That's really where we are in this. Okay, and so the work that was done a lot in my lab on the cryptochrome histidine kinase chimera was done by Lucas van Camier and also Frank Ramirez. My lab has funding from mostly the NIH for different projects dealing with microbiology. And so I'd like to thank you for your time, and happy to answer any questions that you may have.
Thank you for that excellent talk. I'm wondering if you've thought about other species that are sensitive to the magnetic field, and maybe with an evolutionary perspective. There's strains of bacteria called magnetotactic bacteria. They actually precipitate little crystals of magnetite, a lodestone, and then they use it to navigate in the water column to get to different nutrient levels, and they follow the magnetic field lines of the Earth. I'm just curious if there's any possible relationship here or any further investigation that might shed light on these processes.
I think that's a great example of kind of evolution and nature knowing more physics than we do, or taking advantage of physics. And so absolutely, so these magnetotactic bacteria that was described here, what happens is that they form these long chains of kind of iron, and they will align to the magnetic field, and this will lead them up or down. So we think that that mechanism is a little bit different here. And so in some sense, that doesn't lead to a biochemical signal, but rather leads to orientation of bacteria along a magnetic field. What's being described by this radical pair mechanism is actual sensing of a magnetic field that gets turned into something that could affect the nervous system, for example, I should say.
Thank you.
Sure. Yes?
Thank you. That was a wonderful talk. I'd like you to explore the slight difference between quantum sensing and quantum sensitivity. I remember hearing 30 years ago that we routinely make decisions driving based on single photons. And that shows a sensitivity which basically says the laws of physics allow you to do no better. So is that also true for your magnetosensing? Are you constructing something which is actually measuring as faint a magnetic field as anyone could possibly measure?
That is one of the constraints of these systems. So when you look at these relaxation times, the relaxation time of these materials are dependent on how many, so I guess I skipped over this. They're called nitrogen vacancies. So their diamonds are highly ordered, and every once in a while you have a vacancy with the nitrogen in there. And that determines how sensitive of a signal that you can detect. And so quantum sensitivity is absolutely one of the considerations here. The smaller the amount of material, kind of the lower our sensitivity. And also, if the domains are kind of moving around, that also affects our ability to measure these quantum effects.
Beautiful, thanks.
Hi.
Thanks very much. Wonderful talk, exciting. Thank you. A few years, I've been studying the magnetoreception of birds for quite a long time, and a few years back, there was papers out about this receptor or potential receptor called MAG-R. Do you know anything about that? I'm a physicist, and from a physicist point of view, the MAG-R structure looked like a Helmholtz coil, which would make perfect sense for magnetic sensing. Do you know anything about that?
I haven't looked into MAG-R. I have heard of it, but it would be kind of interesting to discuss more about that. And look into it.
I would love to talk to you about it.
Yeah,
if we could get a chance. Thank you.
Thank you very much. It was a beautiful talk.
Thank you.
Easy to understand. I have a question about the sensors. They're quantum sensors, and they would definitely influence the water molecular system in the cell. So have you found some differences in the system brought by the existence of the sensors inside? Because then you'll have, of course, you'll have the effect of sensing, but before that, you will influence your system by those small sensors.
So if I'm trying to understand your question, you're saying that if you put the sensor inside, so this NV sensor inside,
yes.
Does that affect the operation of the signal
The whole
transducers?
system, yes.
That's a great question. We don't know yet, and so that is an area. So we're actively working with these sensors inside bacteria, and we'll ask questions like that.
Yes.
Great question. Yeah,
Thank
I think your question kind of raises this broader question, which is that how do such systems can they be used for signaling within organisms?
Yes.
Yes, exactly.
Exactly.
Yeah.
Yes, thank you.
Hi, thanks for your great talk. This could be a very naive question, but connecting this with some of the earlier speakers today, is it you're looking for a biochemical signal coming out of this sensing that goes on? Is it possible that there is, instead of a biochemical signal, that there's some kind of resonance signal or quantum signal that might be connected to the microtubule resonances?
That's a great question. I think we need to think along those lines a little bit more. I think we're looking at a very specific mechanism. We're looking at unpaired electrons, but there could be more global effects. And kind of how those global kind of electron states could entangle is a great question. And so I guess I would defer to the previous talks in trying to understand that. But I think that's where the state is at and where the gap that needs to be bridged.
Thank you.
Okay, I guess a last question. Yes.
Hi, thank you very much for the talk. What have you thought about using these NV centers for imaging in radiotherapy, where you can actually see how much and where the radiation affected the tumor or the healthy cells around it?
That would be one potential application of this. And so I think others have thought about the use of this in medicine. Absolutely. So the idea is that if you have damaged tissue around, where you have ionizing radiation sources, that's when you're going to have these reactive oxygen species, and one could potentially determine the extent of damage to tissue using this. Great question. Thank you.
Thank you. That was fantastic. So we're going to take a short break, 10-minute break, and we'll start back up at 10:35. Okay, so we do need to get started with the next panelist. So I'd like to introduce to everybody our next on our panel, Nirosha Murugan. Dr. Murugan is a recently appointed Tier 2 Canada Research Chair in Biophysics from Wilfrid Laurier University in Canada. She holds a federally funded PhD in biomolecular sciences from Laurentian University, where she developed patented technologies to detect and halt malignant tumor growth. She was then recruited to the Allen Discovery Center at Tufts University as a postdoctoral fellow, where she developed a successful research program grounded in regenerative biology and comparative cognition with Wyss Institute for Biologically Inspired Engineering at Harvard University. It was during this time that she was also a teaching fellow at Harvard University in the Department of Molecular and Cellular Biology. Now spearheading her own lab in Canada, Dr. Murugan's federally funded research program integrates these works to focus on how biophysical signatures from mammalian systems can be detected and applied to diagnose and reprogram disease states. So, hello, Nirosha. Welcome, and please take it away.
Awesome. Can everybody hear me okay?
Yes.
Awesome. Okay, great. So I will share my screen. So hopefully everybody can see this, and I'd like to thank everybody for inviting me to share our work. So as Paige mentioned, I am here in Canada, and I regret not being able to share my work with you in person. I am here at the Wilfrid Laurier University, and also an affiliate at the Allen Discovery Center at Tufts University. So the main goal of our lab is to really understand how biological information is processed, to ultimately orchestrate how shape and structure is formed within a host organism. And we leverage biophysics by looking at signaling from a bioelectric, bioelectromagnetism, and biophotonic perspective. With skills leveraging in bioengineering, we're really looking at seeing how this information is processed from cells within their own microenvironment and how cells communicate with each other and their neighboring cells to essentially program healthy tissues and how those signals go haywire in non-healthy tissues. So typically, kind of just setting the stage, the typical landscape for cell communication and tissue patterning really is grounded in some form of chemistry. There's molecular control from a chemical or genetic perspective that is well established in terms of that gives us signaling towards normal patterning and how those signals go haywire in mispatterning. And as we all know, the genetic landscape or these gene regulatory networks are convoluted and complex and often not representative in terms of the post-translational modifications that is required to ultimately give us shape within tissues and organisms. So if I were to ask you rhetorically, what shape does this make from this gene regulatory network, I would hard-pressed for you to actually know that this is associated with limb formation. And I mention this because it doesn't really explain the intricacies that's involved in terms of the spatial dynamics and the temporal dynamics that is required to form a complete limb in perfect patterning by just looking at the chemical landscape. And I'm not the only one to make this position in terms of these field level dynamics in development and pathology. There's this excellent review that was recently written to highlight the role of these gradient fields that are responsible and in parallel to some of these genetic landscapes in development. And this excellent review essentially highlights the role of these gradient fields or these morphogenic fields that propagate across the entire organism that gives you these particular fields. For example, you might get a forelimb field, an eye field, a neural tube field. So there's some sort of patterning in terms of the signaling dynamics that's not chemical based, that when they go haywire, you get these, these supernumerary conditions in development. For example, in planaria with multiple heads, or we've shown that just by manipulating these morphogenic fields bioelectrically, we can change the amount of limbs that are regrown back in complex organisms like frogs. And this gradient field, again, which highlights this complex signaling, which up until now we don't really understand where these come from, but we highlight an overencompassing word called the morphogenetic field. It encompasses things like the bioelectric field, this epigenetic landscape, magnetic fields, as we had a great introduction in the previous talk. But now what I want to highlight is that there might be this quantum field that might be regulating signal processes towards patterning in mammalian systems, and these morphogenetic fields may go haywire in pathological conditions. And what I'm going to present for you today is two systems, one in cancer and one of neurodegenerative diseases, that we can leverage these quantum signaling for early detection for these mispatterning states. And before I get there, I want to also emphasize leveraging some of the new techniques that we have within the scientific landscape. So tissue engineering allows us to explore some of these quantum processes, especially in neural architecture, because it gives us a really specific control in how we can shape and pattern these tissues. So what you're looking at here is a review that we've written together to highlight that you can actually control neuroarchitecture in terms of spheroids, modules, manipulating the extracellular matrix in a very, very fine-tuned way to look at how these quantum processes and quantum signaling is actually involved to create organoids and ultimately tissues and full functioning organoids. But that being said, I mentioned that chemical signals are what people have been looking at that regulate the cellular communication. Mike Levin, amongst many others, have also showed that membrane physiology may act like this master regulator that ultimately controls that molecular and genetic landscape and plays a role to this morphogenetic field. So with that being said, I also want to highlight some of these important scientific advances that have been made, that any cell type that you have within your body can be mapped on a scale of membrane physiology. So what you're looking at in the top left corner is that, let me see if I can get my laser pointer, here we go, that, neurons, for example, we know that they're in a very hyperpolarized state, and across this landscape, we can actually cluster and start grouping together cells based on their physiology. And we now know that tumor cells are depolarized compared to other quiescent cells that have more functioning. So this kind of gives us an indication, and it might be that link to the biochemical, the bioelectrical landscape to this quantum that we're trying to explore within biological signaling. With that being said, there is a lot of work on theoretical modeling to show that the ion channel dynamics themselves may actually follow quantum principles. So as many of you know, that these ions are very selective to the channel that they permeate, and that this selectivity isn't just necessarily on this arbitrary lock and key model that we have learned through many high school biology courses. In fact, that selectivity is made probable based on quantum interference, and that some of the water molecules that you have within these ion channels might help regulate what the selectivity filter, so a section of these ion channels that allow certain ions to propagate through. So again, this is an excellent review that was written by Solari that emphasizes that we know that these ion channels might engage in quantum interference to allow for selectivity in these ion channels, but we know that neurons and neural communication is highly dependent on these ion channels. So we're kind of starting to see that link between bioelectric states that are controlled by ion channels. If ion channels are mediated through some sort of mechanism of quantum interference, then there is a probability that we can explore that neural communication might have quantum principles that allow for its information processing, especially within the brain. One other review that I want to emphasize and highlight is this ion channel dynamic, specifically not just in the selectivity of these ion channels, but also in the dynamics. There are specific regions with these ion channels, such as the S4 alpha helix, that is very charged and highly conserved across many different ion channels, and again across many different species. So this might be a location for us to kind of investigate these quantum principles, in the sense that there might be some form of quantum tunneling in terms of the electron dynamics between closed and open states. And I'm not going to get into it in this talk, but there's a lot of evidence to suggest that phantom pain that may not be explained in the classical dynamics of ion channels may be explained through these quantum principles, because they're still engaging these ion channels to give these phantom pains after limb amputations or after nerve injury. So with that being said, this is sort of a good foundation to kind of highlight what we focus in our lab, is to essentially read and rewrite biological states using quantum phenomena. So what I'm going to present for you today is existing literature that uses some of these quantum phenomena to read biological states. So you may have heard of SQUID, or superconducting quantum interference, which uses ultra weak magnetic fields to read brain state. And what I'm gonna, sorry, technical difficulties here. What I'm gonna share with you is the use of these ultra weak biophotons, coupling them to biological state, and how they can be very precisely used to read out not only mispatterned states, but also neural activity. What I won't present today, but what we are also working on in our lab, is rewriting these biological states by leveraging some of these quantum principles. So as I mentioned, the selectivity of these ion channels may follow some of the electron dynamics through quantum tunneling, or what they have shown, the Joseph spin effect, where magnetic fields can actually also alter the electron dynamics through these ion channels, ultimately changing the membrane potential of cells. And we know that when we have these slight modification of these membrane potentials, the whole cell kind of takes over its system by rewriting its state. So we can actually reprogram healthy states into disease states, and also disease states into healthy states, and vice versa. So with that being said, I kind of want just to give a quick introduction to biophoton emissions, or ultra-weak photon emissions. These are very, very weak photon emissions that are exhibited and given off by all living tissue. This can be repeatedly measured over time. These emissions are between about 100 to 1,000 photons per centimeter squared. Ultimately, this equates to about 1,000 times weaker in terms of the sensitivity of the human eye. So there are significant advancements. We've come a long way in optics to actually record these photons, and some of these people have used CCD cameras, PMTs, and now in our lab we're leveraging single photon detectors that they use in aerospace to kind of record these biophoton signals. And we know that the range of these photonic signals are within infrared, and between that range of 200 to 800 nanometers. And many research over the last century have shown that these photon signals are coupled to cell state. So we see this in bacteria very cleanly, that based on their division state, or based on their exponential growth state, we can identify what state these bacteria are in by just looking at their photon signals. So we can see here that Tilbury and colleagues have shown that with just counts of about 10 to the power 3 photons per second, looking at either just the UV or the visible phase, we can actually visible wavelength, we can actually see what phase these cells are in their growth and development. Many people since 2009 have also showed that plants exhibit biophotons, especially when they're wounded, and they also might be coupled to various photonic states internally. If we scale this up to humans, we also see that researchers have used CCD cameras to highlight these biophoton emissions within respiratory conditions as well. I want to, again, tie what we have talked about so far to the previous talk in terms of Albert's talk showing the importance of reactive oxygen species, which seems to be one of the mechanisms by which biophotons are emitted from cells and potentially communicated through this bystander effect, which I'll talk about in a minute. Again, this is a great review that highlights that these biophoton emissions are not just spurious, but also great readouts of what's happening metabolically within cells. You see this, as I mentioned, in very highly proliferative cells like stem cells, in this case, a developing embryo. We see that as you go from the 2-cell stage, as it starts dividing into various components, you see these spikes in biophoton emission. So this is kind of work that was done a long time ago with very rudimentary photonic tools, but we start to see the importance of these readouts in terms of these emissions to biological state. Where I am very interested is trying to understand and find the link to these biophoton emissions to biological state and also the mechanism in which they're formed. So there is a leading hypothesis that the production of intercellular ROS, reactive oxygen species, might be the mechanism in which we're getting these excited states of these species formed and emitted out of these cellular conditions. This is sort of an implication for the radical pair mechanism that has been showcased in the previous talk, which could be the link to the quantumness of the communication within cells. And I'm going to leverage that these photonic signals are not just within the cell itself, but they can be propagated in long distances, potentially for neural communication as well. Myself and many other research, we have some unpublished data to show that by applying magnetic fields to these excited states to increase the reactive oxygen species, we can manipulate the biophoton signals, sort of creating like an amplifier to what's happening inside of the cell. So what I'm going to show you is some work that we've done over the last 6 to 7 years to show that the biophotons are indeed a very good readout of cellular state. So we have cultured many different kinds of cancer cells, placed them on top of these large bandpass filtered PMTs, or photomultiplier tubes. And what we can get is, the first step is that, just looking at media only, so without any kind of cancer cell or cell in there, though there are proteins in there, we're not getting any reliable spikes or alterations across time. But as soon as you start culturing these dishes with cells inside of them, we start to see this pretty chaotic pattern of emission of these photons throughout time. To manipulate this and to actually show the specificity of these photon signals, we have applied a whole slew of inhibitors and activators for cell signaling pathways, reactive oxygen species inducing pathways, and also ion channel blockers to help show that when you systematically do this, we can alter the ultimate biophoton emissions that are being released from these cells. So it gives us some indication that these photonic signals, has been well established throughout years, that they are actually coupled to cell state. And my goal, again, was to get more specificity. What do these biophoton signals mean? How are they actually related to cellular state? So in the subsequent experiments, we kind of do the same exact thing, but this time we try to get the information or the energy within those photonic signals by applying exclusion filters at very specific wavelengths. So as many of the researchers have shown in the past, that these photonic signals are between the UV to the IR range, we've selected these exclusion filters in very specific wavelengths to kind of see, based on cell stress, can we kind of decipher some form of information about what those biophoton signals are. And we've published this quite a while ago now, where we showed that when we induce cell stress to these cancer cells, we get these temporal resolution of these wavelength-specific signals. So throughout a 24 period, by looking at these photonic counts, we see a variation of certain photonic signals in different wavelengths over time. What we're doing now is trying to make sense to what these wavelengths are associated with in terms of that biological process. We know that the very specific cell signaling pathways associated with the mitochondria that are released when a cell is stressed. And now what we're doing is kind of looking at those relationship of those pathways to what these wavelengths are, to kind of find that link between the energetic and the temporal resolution of these biophoton emissions and the cellular landscape from a molecular perspective. But what we have so far is potentially a non-invasive readout of cell state. So especially from a cancer perspective, which we know has a very different landscape in terms of molecular signaling, mitochondrial signaling from healthy cells. So what we have done then is taken biophotonic signatures over 22 different cell lines that were both healthy and non-healthy, and tried to develop a discrimination-based algorithm just by using the wavelengths of biophoton emission to see if we can classify what is cancer and what is not cancer. And the data that we published in Biochemistry Biophysics report shows that we can. And there are very specific filters that highlight that discrimination potential. So what you're looking at in this graph right here, so MDA MB231 is a very aggressive breast cancer line, whereas ATK293s are healthy cell lines. And just by looking at these patterns, we can see that over that stressing period, their behavior is very different from each other, and this behavior can be clustered based on cell type. And we can accurately classify, using a training model, about 97% of all the cell types that we measure using this method. With that being said, we wanted to see how far we can take this. So can we do this in vitro? So what we have developed is a experiment where we had animals that were injected with B16 BL6 cells, which is a highly aggressive form of melanoma. And since we know that biophotons are related to ROS, which is sequestered by the immune system, what we did is we irradiated those same cancer cells and injected them to induce that immune response, but not allow that cancer cell to grow. By UV irradiating them, we're essentially killing the cancer cell, but still, when we inject them into the animal, we're inducing an immune response. And then we have an uninjected control, which it considers our wild type. So this animal will not develop any cancer. So the idea here is we have 3 groups where animals, in 2 of them, they won't develop a cancer, and 1 of them, they should. And we recorded their biophoton emissions immediately after injection and daily for up to 30 days. And what we found that within 24 hours after injection, that we can accurately discriminate. Again, this is a double-blind study, I should mention. About 84% of the cases using very specific frequency bands of the photon emission. So we looked at the spectral density, or so the fast Fourier transformation of those raw photonic signals. And based on very specific frequency bands, we can cluster whether these cells, whether those animals were injected with something cancerous or not. And this holds up to something that we've seen very robustly 7 to 10 days post-injection, where we get about an increased classification accuracy, about 91%. Why this is very exciting is because about after 7 to 10 days, about 15 days, you start to palpate cancers. So at that point, we don't really need an early detection tool. So within 24 hours to about 7 days, when there's no palpable tumor, when there is some molecular dynamics for the tumor formation, these biophotonic signals may be a great readout to kind of for that early cancer detection potential. So this set of data, which I'll summarize, essentially shows us that biophotons, or ultra-weak photon emissions, not just from their raw emissions, but also from their discrimination from their wavelength and their spectral power, can be used potentially for reading out cell state, in this case, cancer versus non-cancer. The next set of data that I want to highlight, essentially using these same biophotons, but for information processing within the brain. I'm not the first one to postulate this, but there's many different reviews that have suggested that maybe the axons within neurons within that propagate information throughout our brain may act like optical fibers or optical channels. And interestingly, that we know that biophotons may actually be traveling along these axon bundles, carrying information to and from, that may actually be involved in affecting bystander neurons in terms of that high energy photon emissions that are being released. On to that point, there are some great reviews and theoretical modeling by our colleagues, Christoph Simon and his group, which have suggested that the fact that the neurons might allow for light conduction may give us some insight to look at the brain as a quantum computer potentially, and have suggested that the way that the myelin covers the axon can give us some potential for backward transmission of information. And we see that in classical information processing within the brain, the electrical conduction is only unidirectional along the axon, and potentially these photons can allow for backward propagation, therefore enhancing some of the information processing within the brain. Relating to the quantumness of this kind of relates to hypotheses around spin chemistry, which Albert gave a great introduction into, where we showed that we see this, in animals that are involving cryptogame, for example, in migratory birds. But we also know that the human brain is magnetosensitive. Though we do have locations of cryptogame, what we postulate is that the axons, which you see in this image themselves, propagate mitochondria from its cell body down to the dendrites, might be sources of these biophotons that are not just propagated along from as like an information highway, but are also actively involved in information processing. So this is a great review that was done that kind of highlights the hypothesis of this. And what we're doing is now we're taking this to the next step to actually record this in real time. So we have two objectives that we're currently undergoing with our collaborators from Calgary, Waterloo, and France, through actually bioprinting neural-inspired waveguides. So we're going to recapitulate the material properties of axons and print them in very specific orientations to see how those axons or those bio-inspired axons propagate light, and can we actually read out information from those states. Onto that point, we're kind of taking it to the next layer by looking at the quantum sensing ability of these brain-derived biophotons, or also weak photon emission. So this is work that's been funded by Optica that we hope to present in the near future. But to that point, what I'd like to present for the last little bit is to show that these biophotons are actually involved in neural communication and can be used in human settings for brain state readouts. So what we are really focusing in, because we are also a cancer lab, we've noticed that patients who are undergoing chemotherapy have experienced something called chemobrain, which is a debilitating state where they have memory loss, anxiety, and all sorts of cognitive impairments, which often can last much longer than traumatic brain injuries and have some of overlaps with Alzheimer's disease. So what we propose is that we can actually leverage the use of biophoton signaling, coupling them with neuropsychometrics and electroencephalography, so using that multimodal approach of looking at biophoton emission and electrical emission using EEG to get a holistic readout of the brain. And potentially, if biophotons are an early readout of brain state, they can actually give us an early readout of chemobrain, where clinicians then can intervene much earlier to alleviate this problem. So as a proof of concept, we've designed a study with 120 patients, ranging from 18 to 67 years old. All patients undergo several neuropsych assessments, profile of mood states, and states for mild traumatic injury. We also engage or apply an EEG cap onto the participant, along with our custom developed photonic tool, which records records biophoton emission from the temporal lobes and the occipital lobes in this study. And in this pilot study, what we have done is developed a very, very proof of concept study to show that the brain actually changes when there are auditory stimuli present. So what you're looking at here, what the participant is engaged with is a 2 minutes eyes open baseline, 2 minutes of a very, very annoying auditory stimulus to kind of engage their temporal lobe, and then again, eyes open, eyes closed condition, so that we can actually visualize the changes within their brain state that has been well validated in using EEG. So since we're kind of exploring new areas, what we have shown here is that the EEG displays what we're supposed to be seeing. So we're seeing alpha power in the occipital lobe as a function of eyes closed, which is well established, so that we know that our design is measuring what it's supposed to be measuring. So that's great. And if we look at our photonic signals now, which is where the novelty lies, is that we are getting brain relevant photon emission. So what you're looking at here is background PMTs that are placed near the participant, but not near their head. It's actually located close to their leg as they're sitting down. We see no fluctuations in that photonic activity, but sensors that are placed within their occipital lobe or their temporal lobe, we start to see these kinds of fluctuations that potentially might give us insights to the brain state. So what we're doing right now is kind of analyzing this data and actually trying to figure out how to analyze this data. So one way that we've highlighted is that we've looked at recurrence plots that looks at the deviation of these photon emissions over time to see if there's any kind of photonic signature that is relevant to their brain state. And when we look at just the background signals of those photon emissions, we see no difference in terms of the photon signatures across the experimental conditions, so auditory versus eyes closed or eyes open. If we look at just our occipital and our temporal, we start to see that there are no similarity across their phases. So this is just another analysis method to show that those raw photonic signals might have information in them that might be able to discriminate between brain states. And that's, again, another layer of analysis that we've done where we've taken the raw photonic traces, and we actually found that there are 5 different kinds of subtypes of signatures within this to kind of give us differences between background, the occipital sensors, and the temporal sensors. And, again, this is some pilot analysis that we've done to actually showcase that using this photon signals, we've developed a way to look at the variability within those signals. And based on eyes open before the music, during the music, or that auditory stimulus, and after, we can actually see that the background PMTs of photon emissions don't actually change across those conditions. And it is expected to see, within the occipital lobe, that we do see those consistent increases in photon emission throughout time. And what's very exciting that we see is that we only start to see a difference within the temporal sensors only when the participants are engaged with some sort of auditory stimuli. Again, this is work that we're still pursuing to try to find the important characteristics associated with those signals that are actually going to give us a readout of brain state. So I'm just going to summarize what we are doing and what we have shown so far, and that these emissions that we see from electrical signals, magnetic or biophotonic, that what's the most important isn't the fact that they are emitting these signals, but that's great, but the pattern in which they emit these signals might be a great readout of the chemical landscape or the molecular landscape that's happening within the cells. And if we listen or if we engage with these messages, we can digitize these signals and apply them back in a way that we can resonate with these molecular signals in a more fine-tuned approach, so we don't get the sporadic effects that we see throughout time. In terms of our study, what I've shown you today, what I'm hoping to emphasize, it is actually very important for the community to develop a set of variables that is consistent to characterize these photonic signatures, especially as we move towards something complex such as the brain in terms of brain signaling, neural readouts, and brain state. And what we're looking to do right now is that we are engaging these kinds of platforms to look at intrapersonal and interpersonal readouts as we multiplex our devices with adding the electrical landscape, as you've seen today, with EEGs, and we're also multiplexing this with MEGs to look at, how much of that neural information is actually electrical, how much of it is meaningful magnetic information and photonic. And hopefully soon we'll be able to share that with you at upcoming conferences. So with that being said, I want to thank our lab for doing some of this work and also our funding support. So with that being said, I'll leave it to you for any questions.
Thank you so much, Nirosha. That was so fascinating. That was wonderful. We have time for a couple of questions. So please go ahead.
Hi, can you hear me okay?
Yeah.
Super. Thank you for that fascinating talk. I really enjoyed that. As you were talking, I was reminded there's a familial variant of ALS or MND in which superoxide dismutase 1 is mutated. It occurs to me that that might be quite a good neurodegenerative disease candidate for the kind of work that you're doing. Is that something that you guys have considered?
Not particularly in that pathological state, but we are looking at superoxide dismutase mutations and how they are involved with photon emission. We know that it's very involved in magnetic applications. So we're looking at that intersection between how much of this is just raw photonic signals, how much of it is it magnetically involved. So something that we're working towards.
Thanks.
Please.
Yeah, thanks for a really fascinating talk. I have a naive question. You mentioned that the range of wavelengths of biophotonic emissions is relatively narrow, and it seems like it matches somewhat the visual spectra that our retinas are sensitive to. Maybe a little bit more in the infrared range, though I understand that our retinas actually are sensitive to ultraviolet were it not for the filtering effects of our lenses. My question is, evolution could have selected for a very broad range, or at least broader than what we're finding. So why do you think that is? Is there some reason why this range is so narrow?
Oh, that's a great question. I mean, I love that you emphasize the evolutionary approach to this. We know that some of these protein interactions is selected for within these wavelengths, but I'm not exactly sure as to why this very specific narrow band pass or this very narrow wavelength for our mammalian processing. Great question, though.
Okay, thank you.
Okay, and last question.
Thank you very much for the presentation. Before you end, could you put your contact information slide back up there so I can get a screenshot of it? And my question is this, in your lab or your associates, have you been examining the historic role of indigenous and ancient healers and the whole philosophy of generating light, either the shamanic practitioner generating the light to assist the patient to generate internal bodies of light themselves as healing modalities?
Yes, great question. I've done some of that work in the past by measuring biophotons coming from healers. We've had various kinds of projects where these healers were emitting biophotons as a function of their treatment on cancer cells. So we've done some of that work, and we're potentially starting a collaboration with someone to look at the healer-healee dynamics to see if there's any kind of EEG synchronization or photonic synchronization during that process. So potentially upcoming work in the near future.
Great. Thank you so much, Nirosha. Okay, so moving on to our next speaker, I'd like to welcome Manisha Patel. She is a doctoral candidate at the University of California, Santa Barbara. She received her bachelor's in physics from Stanford University. Her work focuses on understanding non-classical nucleation pathways of calcium phosphate for the purpose of controlling biomineralization and testing Posner-mediated quantum brain theory. Manisha, please go ahead.
Hi, everyone. Thank you so much, Paige, and all the other speakers. This has been really fascinating. I'm so sorry I can't be there in person. I had a flight cancellation yesterday, so I'm hoping that I'll be able to make it in person tomorrow. Fingers crossed that the flight won't be canceled again. Okay, so I'll just get started then. Let me get my screen organized. So I'm going to be presenting some recent work from the Quantum Brain Collaboration headed by Matthew Fisher here at UCSB in Santa Barbara, where we've been focusing on trying to elucidate non-classical lithium isotope effects. And before getting started with the work, I'd just like to acknowledge my collaborators, in particular Josh Straub, who's a recent PhD graduate with whom I worked very closely on this project, as well as my two advisors, Matthew Fisher and Matt Helgeson. Okay, so I'll start off by first providing some context for why we're interested in lithium isotopes. Lithium is of interest in medical research due to its common pharmaceutical applications in treating mental illnesses, such as bipolar disorder. However, the exact mechanisms for how lithium interacts with the brain are not very well understood. And interestingly, certain biological systems have shown a differential lithium isotope effect between lithium-6 and lithium-7, which are two stable isotopes of lithium. For example, a few years ago at UCSB, a collaboration study looking at the manic behavior in rats demonstrated that ketamine-induced behaviors were differentially impacted by two lithium isotope treatments, where lithium-6 suppressed manic behavior significantly more than lithium-7. That figure is shown here. And more recently, it was shown by some of our collaborators at TU Munich that lithium isotopes have a differential effect on the sequestration of calcium within mitochondria, where the timescale for release of calcium is notably different when isolated mitochondria are treated with either lithium-6 or lithium-7 based buffers. And it's well known in the literature that calcium is stored within mitochondria in the form of calcium phosphate, likely in the amorphous calcium phosphate phase. So at UCSB, we wanted to ask whether these lithium isotope effects that were observed in mitochondria, as well as other biological systems, might actually be arising from a differential effect on calcium phosphate formation and stability. So we're attempting to reduce this problem that originated in biology to the level of physical chemistry. So what could possibly explain this differential isotope effect that appears at a range of scales? So we take a very physicist approach of just reducing things down to first principles by considering the basic physical differences between lithium-6 and lithium-7. So as we know, isotopes share identical electronic states as well as the same number of protons, but they differ in the number of neutrons in the nucleus. So this will give a mass difference between lithium-6 and 7 of around 16%. However, in aqueous solutions, these ions will always be surrounded by a shell of water molecules. So in fact, the mass-related parameter that really matters when analyzing aqueous solution behavior is this aqueous diffusion coefficient that I'm showing here, which will account for the hydrodynamic shell surrounding free ions in solution. So this diffusion coefficient was measured to be nearly identical between lithium-6 and 7. They differ by only about 0.6%. So this would also imply that the local water structure surrounding lithium-6 and 7 ions are nearly identical between these two isotopes. So we find very limited explanatory power in a mass-based sort of kinetic phenomenon or a local water structure related phenomenon that could explain lithium isotope effects. However, the isotopes do differ notably in their nuclear spin, where lithium-6 has a nuclear spin 1 and 7 has nuclear spin 3/2. It's shown here. And they also differ notably in their gyromagnetic ratio, which will determine the strength of dipolar coupling between spins. And additionally, their quadrupolar moments are considerably different, where lithium-7 has a much stronger quadrupolar moment than lithium-6. And this actually determines the strength of coupling to local electric field gradients. So while lithium-6 and 7 are not necessarily expected to have different aqueous solution chemistries from a classical perspective, their nuclear spin properties are indeed quite distinct. So with regards to lithium isotope effects seen in biology, there's not really an obvious explanation here coming from a classical chemistry perspective. And we're motivated to think about their nuclear spin properties more carefully. So now we can take a step back and ask ourselves what's missing from the picture and how might we be able to build a theory explaining this phenomenon that actually accounts for nuclear spin properties. And there's sort of three key ingredients that I'll discuss here. First is a direct treatment of the nuclear spins and their distinct properties. The second is a full consideration of quantum indistinguishability and the consequences of that. And lastly would be some sort of quantum to biochemical to biological transduction pathway, which I think a lot of people have been thinking about and discussing in this workshop. And as I'll demonstrate on the following slides, our project attempts to address and kind of tie together these three criteria. So again, starting from first principles, we can identify a well-isolated quantum degree of freedom to be nuclear spins, which are ideal given their relatively weak coupling to the environment compared to other spin properties. And just considering the most common biological elements and their nuclear spins, we find only 3 possible elements that are ubiquitous enough in the body to play some non-trivial role that also have a spin-active nucleus, and that would be the proton, nitrogen, and phosphorus. Now, protons are perhaps a little bit too ubiquitous in aqueous environments and would likely have very uncontrolled interactions compared to the other 2 options here. So just considering nitrogen and phosphorus, nitrogen has a nuclear spin of 1, which will decohere very strongly with electric field gradients, whereas the phosphorus has a nuclear spin 1/2, which will only be weakly decohered by magnetic fields. So this leaves the phosphorus nuclear spin as our preferred biological qubit, if you will. Now, a problem arises here given the fact that in biological environments, phosphorus is housed generally in phosphate ions, which are usually protonated, which leads to faster spin relaxation. And a solution to this problem would then be some sort of biological cation that can easily outcompete with protons in binding to phosphate. And calcium is well known to be such a cation, as calcium phosphate species are found throughout the body. And it also has a spin-inactive nucleus, which makes it an attractive candidate as it won't decohere with phosphorus nuclear spins. Now we can consider a molecular calcium phosphate vehicle that may carry isolated phosphorus nuclear spins in a protected manner. And the Posner molecule appears to be an attractive candidate in the literature. So basically, a Posner molecule is a single nanometer size calcium phosphate nanoparticle that plays a key role in calcium mineralization processes, both in vivo and in vitro. And importantly, the Posner molecule has been predicted to have very long phosphorus spin coherence times on the order of minutes to days. And this is due in part to its small size and also rapid tumbling rate in water. And another key feature of the Posner molecule to consider is its rotational symmetry. So it has what's called a C3 rotational symmetry along the central calcium axis that's shown in the dotted line here. And as I'll show on the following slide, this symmetry plays an important role when considering the full implications of quantum indistinguishability. So I'll emphasize now that phosphorus nuclear spins are fermionic, indistinguishable particles. So due to fermionic statistics, we know that when 2 phosphorus nuclei are exchanged, their wave function has to transform antisymmetrically. So for the particular geometry of a Posner molecule, where there's a 3-fold rotational symmetry that will cyclically permute these 6 phosphorus nuclei, it was shown in this paper in 2018 that a rotation will induce a phase factor on the nuclear spin wave function for the molecule, introducing what's called a pseudospin value corresponding to these 3 rotations. Now, the details here are not terribly important for the talk, but you can kind of think of pseudospin as a measure of the collective nuclear spin states under a cyclical rotation. And now, given that the total wave function for this molecule will consist of a product of nuclear spins, nuclear rotations, as well as electronic states, this antisymmetry requirement arising from indistinguishability imposes a coupling between the orbital angular momentum of the molecule and the pseudospin quantity that's indicated by tau. And the derivation of this relationship is shown in the paper below, but I'll just state the result here for the particular C3 symmetry, where we basically have a direct relationship between angular momentum given by L and the pseudospin quantity tau. So to summarize the result just in plain words, we find that the orbital motion of Posner molecules can be constrained by the collective nuclear spin states of the phosphorus nuclei. So this result leads to the development of the theory called quantum dynamical selection, which relates this coupling between pseudospin and orbital motion of a molecule to actual binding rates between small symmetric molecules, such as Posner's. And the central conjecture of quantum dynamical selection is that bond formation for small symmetric molecules requires a zero angular momentum state, which in turn relates to a pseudospin sum of zero for a pairwise binding event. And that's just kind of written in mathematical terms here, where we have a requirement that the total pseudospin of two binding molecules must come to zero in order for molecules to actually form bonds. And now to illustrate this effect more clearly, we can consider a thought experiment where we have two populations of Posner molecules or any other small symmetric molecule that satisfies these criteria. And let's say they have two pseudospin populations of up and down, or +1 and -1. And now in a pairwise binding event, in the case where the pseudospin sum comes to zero, where let's say you have a +1 and a -1 molecule attempting to form bonds, relative rotations may momentarily come to zero due to this requirement between the pseudospin sum being zero and the orbital angular momentum being zero, and that will allow for bonds to form. However, if the pseudospin sum is non-zero, relative rotations are not able to cease momentarily and bonds are not able to be formed. So this theory provides sort of a transduction pathway between quantum properties, the proposed quantum properties of Posner molecules, and sort of nanoscale biochemical reactions. Now returning to this ingredients list, we now have satisfied the first two criteria here, a direct treatment of the nuclear spins, and then a full consideration of the consequences of quantum indistinguishability. And we've also identified a quantum to biochemical pathway. So the last piece that's missing here is a full, a biological platform on which this mechanism can play out. So mitochondria have been discussed a lot in this workshop, and they also provide an attractive biological transduction system for our project. As I said, it's well known that mitochondria play an important role in storing excess calcium in the cell in the form of calcium phosphate. So they play a very key role in modulating the amount of free calcium that's available for cell signaling, such as neurotransmitter release of calcium ions. And in neurons in particular, mitochondria exist in these highly dynamic networks that have been proposed to play a role in information processing in the brain. So these networks provide a potential platform that we try to keep an eye on for manifesting quantum effects at biological scales. So now I'll actually be able to discuss our experimental work done in this effort, where we attempted to probe potential quantum properties of Posner molecules by measuring their downstream effects on the nucleation and aggregation behavior of amorphous calcium phosphate. And as a tool for measuring these downstream effects, we can bring in lithium isotope substitutions into calcium phosphate that will differentially disturb phosphorus nuclear spin states in search for some sort of differential lithium isotope effect here. So this project really ties in the Posner-mediated quantum brain hypothesis back in with the lithium isotope effects that sort of served as the original puzzle for the project. And to make the hypothesis a little bit more clear, we know that lithium-7 couples more strongly with phosphorus nuclear spins than lithium-6, which would imply that lithium-7 shortens phosphorus nuclear spin coherence times on a faster scale than lithium-6. So we can then ask if this differentially influences Posner binding rates as predicted by quantum dynamical selection, and thus having a downstream effect on nucleation and aggregation of amorphous calcium phosphate that we can more easily measure in solution. Now to provide more context for the solution behavior of amorphous calcium phosphate, this schematic is showing the solution phases of calcium phosphate and all of their transitions. And the schematic is based on a whole body of work in the literature that suggests this very complex multi-step nucleation pathway involving multiple metastable phases. So I'll just try to go through each of those steps here. So we have, in the early stages of ACP growth, we begin with just dissolved ions of calcium and phosphate. And then over very, very short time scales, these ions will stochastically form what are called prenucleation clusters at the single nanometer length scale. And there have been multiple prenucleation clusters suggested in the literature, including calcium triphosphate clusters, which have protonated phosphate ions, as well as the Posner molecule, which we've been focusing on. These prenucleation clusters will then very rapidly aggregate to form larger amorphous calcium phosphate particles, which will then undergo colloidal aggregation into larger structures. So our central question then is whether or not lithium incorporation into this prenucleation cluster phase can influence the formation of amorphous calcium phosphate, and is that effect similar between the two isotopes of lithium? Okay, so we use a technique called dynamic light scattering to measure the downstream effects of aggregation of ACP particles into larger colloids. And I'll be situating our results in sort of pH versus calcium phase space. And the diagram is shown here on the left, where we have a fixed concentration of phosphate for our solutions. And the red region here, which I'll be exploring, corresponds to the colloidal aggregation phase for overall solution behavior for calcium phosphate. So the basic setup for our experiment is shown here, where we shine a laser on aqueous solutions of calcium phosphate that are undergoing colloidal aggregation, and then monitor the growth of particle sizes, as well as just the raw scattering intensities over a period of 5 minutes. And the two figures on the right here are just showing an example data set, where the top is showing the growth in particle diameter over time, and the bottom is the corresponding raw scattering intensity. And we performed this experiment at a range of solution conditions and found quite an interesting trend. So here at very low physiological pH and 7 millimolar calcium, we find that the particle sizes, as well as count rates for the scattering intensities for the two lithium isotope doped ACP solutions are exactly identical. And similarly, at slightly higher pH, the particle sizes, as well as count rates for lithium 6 and 7 doped ACP are again identical, and we find no lithium isotope effect. However, within a very specific pH window in between these two limits, we find a very interesting phenomenon where the particle sizes in each case are identical, but there's a notable difference in the scattering intensities that are promoted. Where lithium 7 doped ACP solutions promote a higher scattering intensity than lithium 6 doped ACP solutions. And we found a very similar effect manifesting at lower calcium concentration at 5 millimolar calcium, where again, lithium 7 promotes a higher scattering intensity than lithium 6 with identical particle sizes and within a very specific pH window. And interestingly, as we go to even lower calcium concentration at 3 millimolar, these isotope effects diminish very notably. And in the region that we explored, we found only a very weak difference in scattering intensity over a very small pH window, shown here. So this figure is just summarizing the region in pH versus calcium phase space where this lithium isotope difference in the scattering intensity manifests. And it's interesting to note that the pH window for this isotope effect is most pronounced at a very particular calcium to phosphate stoichiometry of 5 to 2. That can kind of give us a clue for which of the various prenucleation clusters might be playing a role in this isotope effect. And it's also interesting that the isotope effect seems to diminish on either side at both lower and higher calcium to phosphate stoichiometries. Now, how do we actually interpret the difference in scattering intensity? So in the regime of particle sizes that we're looking at, we know from Mie scattering theory that the intensity will scale linearly with particle number or particle concentration, and then quadratically with the particle size, as well as this delta n term here, which is basically the difference in index of refraction between the ACP particles in solution and the surrounding water solvent. And we measure the particle sizes to be identical using dynamic light scattering. So we know that the difference in scattering intensity isn't coming from the sizes. And the index of refraction is dictated by electronic states, which are going to be identical between lithium-6 and 7. So we can cross out this term as well. And we're left here with a direct scaling between the scattering intensity measured in DLS and the particle concentration. So we can conclude here that within a very specific region of phase space, lithium-7 is promoting a higher particle concentration of ACP colloids than lithium-6. And I'd just like to emphasize here that this effect is actually the opposite of what we would expect from a mass-based kinetic isotope effect. So typically for these solutions, heavier isotopes would slow down aggregation kinetics, and we would expect lithium-7 being the heavier isotope to promote a lower particle concentration than lithium-6. So this is a mass-based, the form of mass-based kinetic isotope effects that we've observed for other isotopes. And it's the opposite for the lithium isotopes that we've been seeing here. So now that we've established a lithium isotope effect on the sort of downstream stage of ACP growth, we can try to backtrack along this schematic to try and determine at which stage the isotope effect is actually manifesting. So we measured solutions without any calcium of just pure phosphate and either lithium-6 or lithium-7 chloride using phosphorus NMR, and found very similar traces, which would indicate that the lithium-6 and 7 are interacting with just pure phosphate in a very similar manner. And also just bringing back this aqueous diffusion coefficient, which would imply that lithium-6 and 7 are not locally structuring water in a very fundamentally different way. So we can conclude here that there's not likely a huge isotope effect just in this sort of free dissolved ion phase and the pre-nucleation step. Now considering the later colloidal aggregation stage, we didn't find any difference in the location of the aggregation phase boundary as measured using UV-Vis, nor did we find any difference in the actual incorporation of lithium into ACP particles using ICP mass spec. And also we measured using phosphorus NMR actual full, the same solutions that we were studying using DLS, and didn't really find any significant difference in their phosphorus NMR traces. So this all indicates that there's not likely a significant lithium isotope effect that's manifesting right at this stage of colloidal aggregation that would be indicated by some sort of differential incorporation into that stage. So we're sort of left with this pre-nucleation cluster stage where lithium incorporation into these clusters, or perhaps interaction with these pre-nucleation clusters, may be causing an isotope effect that we then measure downstream. And to confirm the feasibility of this idea, we performed spin counting experiments on lithium-doped ACP powders to actually measure distances between lithium and phosphorus nuclei. And this distance was found to fall between roughly 3 and 4 angstrom, which would indicate the possibility that lithium is actually incorporating inside of these single nanometer pre-nucleation clusters at length scales that would be relevant for nuclear spin interactions to play a role. So I'm just going to emphasize this figure here again, showing the key differences between lithium-6 and 7 that lie in their nuclear spin properties. So any incorporation of lithium isotopes into pre-nucleation clusters like Posner molecules would then differentially impact phosphorus nuclear spin states within that molecule. And as I mentioned previously, quantum dynamical selection would then predict that the rate of aggregation of pre-nucleation clusters into larger ACP particles would then be different for lithium-6 versus lithium-7 doped prenucleation clusters. And just to make that last point a little bit more clear, this figure is showing sort of an illustrative time series of pairwise binding events between Posner molecules as well as lithium-6 or lithium-7 doped Posner molecules. And in this illustration, all of the binding events shown in red have non-zero pseudospin sum. And as shown for a period of time, these Posner molecules are going to be unable to bind due to their non-zero pseudospin sum, as predicted by quantum dynamical selection. However, after a certain coherence time, tau, the phosphorus nuclear spins will then decohere, and the pseudospin limitation predicted by QDS will then be lifted, where the limitation on the rotational motion no longer applies, and binding can then proceed classically. And now lithium nuclear spins will decohere with phosphorus nuclear spins if they're actually incorporating into these prenucleation clusters, which then shortens the coherence time, tau, allowing for more classical Posner binding events to occur. And since lithium-7 interacts more strongly with phosphorus nuclei than lithium-6, the coherence times for lithium-7 doped Posners would be shorter than for lithium-6 doped Posners, which would then result in a higher concentration of ACP particles downstream. And lastly, I just want to wrap it up here with a very brief discussion of how this in vitro result might be relevant to the in vivo mitochondrial lithium isotope effect that I had mentioned at the beginning of the talk. So our collaborators at TU Munich expanded their study of lithium isotope effects to look at both liver and isolated brain mitochondria. And in liver mitochondria in particular, they found that lithium-6 stabilizes mitochondrial calcium phosphate more significantly than lithium-7, where mitochondria that were treated with a lithium-6 based buffer underwent what's called permeability transition at a much later time scale than mitochondria that were treated with lithium-7 based buffer. So that would indicate greater stability of the calcium phosphate pool within mitochondria when they're treated with lithium-6 versus lithium-7. And we suggest an explanation here that lithium-6 might be encouraging fewer large granules or particles of ACP, a lower concentration within mitochondria, which would thereby induce less stress on the mitochondria compared to lithium-7. And perhaps that's an explanation for why lithium-6 is suppressing permeability transition as compared to lithium-7. Okay, so I'm just going to wrap up the main points here, and then I think we'll have some time for questions. So we discovered a novel lithium isotope effect on amorphous calcium phosphate nucleation and aggregation in vitro. And this differential lithium isotope effect appears to manifest during the formation of ACP colloids, where we can clearly measure it in downstream colloidal aggregation, but we provide some evidence for why that actually might be manifesting at this prenucleation cluster stage. And then lastly, this in vitro result offers a potential explanation for various lithium isotope effects that have been seen in biological systems. So that's all I have for content. I'll leave it to Paige for any questions from the audience.
Okay, great. Thank you so much, Manisha. That was such an interesting analysis of the lithium isotopes. So we have time for a couple questions. I wanted to jump in and ask, so what would be the consequences of sort of patients that are being treated with lithium? What is the conclusion that can affect their treatment?
Yeah, so there's reason to believe that lithium-6 might actually have fewer side effects than lithium-7. It's been found, for example, one of the, there's multiple side effects that result from receiving treatment, lithium treatment for bipolar disorder in particular. And one of the interesting side effects that we've been thinking about is tooth decay and osteoporosis. And we believe that might have something to do with how lithium is affecting the stability and mineralization process of calcium phosphate that is affecting bone growth. And given that lithium-6 seems to be able to stabilize smaller particles, essentially slow down the aggregation of calcium phosphate and the overall mineralization of calcium phosphate, it may have a differential effect on how it incorporates into bone and then having an impact on things like tooth decay and osteoporosis. So I would say overall, it's not very clear from these results which of the two isotopes would suppress or enhance side effects in impacting bone growth. But given that lithium-6 is far less abundant, patients would be essentially being treated with a purely lithium-7 treatment. So it would be interesting to see if lithium-6 could reduce some of those side effects.
Okay, that's great. And then one more quick question. Are there any other elements, molecules sort of on your radar that might have a similar mechanism?
Yeah, so magnesium in particular is something that we think about. Magnesium phosphate has parallels with calcium phosphate aggregation and also plays a role in terms of phosphate sequestration in mitochondria. And there's reason to believe that magnesium phosphate prenucleation clusters have similar symmetries to Posner molecules. So that's something that we've been thinking about as an alternate for sort of a molecular vehicle that could house phosphorus nuclear spins. That has a lot of parallels with calcium phosphate.
All right, great. Thank you so much. Thank you for joining us today, Manisha. And so we will jump forward to our final presenter today. I'm excited to be introducing Philip Kurian with us. Dr. Kurian is a theoretical physicist, researching scientist, and essayist, serving as principal investigator and founding director of the Quantum Biology Laboratory at Howard University. Beginning his career as a math teacher in North Philadelphia and completing a stint at NASA Goddard Space Flight Center before graduate school, Dr. Kurian is now the recipient of fellowships, grants, and awards from the Alfred P. Sloan Foundation, U.S.-Italy Fulbright Commission, Guy Foundation, National Science Foundation, and the National Institutes of Health. His laboratory studies how collective and cooperative quantum behaviors can explain biological phenomena at the mesoscopic, organismal, and clinical scales, including in neurodegeneration, cancer, and human consciousness. In 2021, Dr. Kurian was appointed as a lead expert for the National Academies of Sciences, Engineering, and Medicine workshop on quantum-enabled sensing and imaging for biology. In 2022, he became a Simon Scholar and Senior Fellow at the UCLA Institute for Pure and Applied Mathematics. And in 2023, he was selected as a Fellow of the UCSB Kavli Institute for Theoretical Physics. His essays on science, human knowledge systems, empire, and spirituality have appeared in various media outlets, including the Los Angeles Review of Books, Granta, and Plough. Thank you for joining us today, Philip.
Thank you so much, Paige, and thanks to the organizers and Stuart for inviting me and for all the great speakers we've had in this session. Can everyone see my main screen, or are you seeing my presenter slides?
Looks good.
Okay, thank you. So today I'm going to talk about one line of thought in my group that we've pursued over the last decade, and you can find some of the references here from 2017 and 2019. That's work with Travis Craddock, and just recently published on Friday, live in the Journal of Physical Chemistry B, we were selected as an editor's choice pick by Science Magazine. So I'm going to focus on that work and the line of thought leading up to it. But first, I want to give gratitude and thanks to the following people who made that work possible. These are postdocs in my group, as well as an undergrad intern and our collaborators. in Italy and Switzerland, Majed Chergoui's group at EPFL in particular, as well as this work was supported by the Guy Foundation in the UK and leadership computing facilities at Argonne National Lab. At the end of the talk, hopefully I'll get to a new project that we're building off of this one that just published. A postdoc, Suyash Bajpai, and another not listed here, Sangeeta Sengupta, are working on that in my group, but we're also collaborating with Michael Levin and Nirosha Murugan, who is at Wilfrid Laurier, if you heard her earlier in this talk, but has an affiliation at Tufts as well. And that's supported with our Japanese colleagues, Masashi Ayono, by the Alfred P. Sloan Foundation Matter to Life program. So, for those of you who haven't been listening to the earlier parts of this talk, I'll quickly talk about why quantum biology is so diverse of a discipline and why there's so many different roads to arrive at answers in the field. So I'm a theoretical physicist, but as you've heard from those who've come before me, there's a lot of different perspectives in this group. Theoretical physicists like simple models of absorption and emission. So the simplest quantum model is a two-level system, something with a ground state and an excited state. Real molecules don't actually look like that, but this is often a really good approximation. And so it's important to remember, I'm a fan of John Archibald Wheeler's quote about reality. He wrote in this 1983 essay, Law Without Law, that our physical models are really an elaborate papier-mâché construction of imagination and theory fit between a few posts of clear observation. And that's really what we're trying to do, because we know that reality is not physics and physics is not reality, but hopefully our models get closer and closer to reality. So when we approach biology, I actually found out that there's not just spherical cows, there are spherical birds like this. And you can actually see that as we deal with more realistic living systems, we get messier and more nonlinear and more non-equilibrium and more non-adiabatic, where we can no longer assume that things are nicely separated and things operate in thermal equilibrium. And this is life, right? So the interaction of light and life poses all these new physical scenarios in which we have to deal with the complications of these challenges. So this puzzle of long-range spatio-temporal order in biology is a real puzzle, because we have to go from the quantum vacuum. The quantum vacuum is the background of all living systems in all of our universe. Constantly, photons are coming into and out of existence from this quantum vacuum. But this is a scale that we normally don't associate with biology, right? We associate the scale more of hertz and milliseconds and microseconds with what's happening in biological systems. But how do we span all these scales of order from the quantum vacuum all the way to living systems? That really is the challenge. And as you can see here, those photons that are coming into and out of existence will play a role in the work that I'm presenting today. So this is the title of that work. So we're going to talk about a concept called superradiance. And superradiance is a collective quantum optical effect. So it's not a local effect. It's an effect of large collectives. And so if you take anything away from this work, it's not a local effect like in Forster resonance energy transfer or in other systems where we're just looking at single molecules. But this is actually a robust quantum effect that occurs only with large system sizes. So it's a very counterintuitive sense for people who've been told that large systems at thermal equilibrium can't be quantum. So this is a really important result because it suggests that, in fact, there's a whole range of biological architectures that may be collectively in the large size limit quantum. So let me show you. Uh-oh.
Uh-oh. So Philip, if you can hear me, you've frozen up on us. I'm not sure if it's...
Can you,
oh.
Can you hear me okay?
I can hear you, yes.
Okay.
Your screen looks frozen.
Yep, it looks like it's frozen. 1 second. Okay. Okay.
Still there?
Yeah, I'm still
Okay.
here. Apologies. Looks like
Is it
I may have to.
reboot?
Yep, I may have to reboot. I really sorry about this.
No problem.
Okay. Give me 1 second.
Sure.
Thank you. Hi, Paige. Can you hear me? Can everyone hear me?
We can hear you.
Okay.
There you are. Yes.
Great. Thank you. Sorry about that.
No problem.
Let me go back here. And I will try to do it on a single screen to avoid any other challenges. Okay. So let's start back where we were. Okay. Is that okay? Can you see everything?
Yes, it looks like it's
Paige.
in presenter view.
Oh, it's in presenter view. Okay. All right. Let me try it here. Okay. Okay. Is that better?
Looks great.
Okay, thank you. So this is two of the postdocs in my group who are the first authors on this work, Nathan Babcock and Gustavo Montes Cabrera, who is now at the University of Guadalajara in Mexico. So what do these large architectures look like in different systems? So you've heard about microtubules, but microtubules, of course, form other architectures. They form microtubule bundles in neurons. They form the centriole. They're also active in the axoneme and cilia and flagella. But there are networks of these tryptophan that occur outside of microtubules in actin filaments and photoreceptor complexes like UVR8. In plants, it's actually a funnel made of tryptophans. There are transmembrane proteins, and even in whole viruses, there are these networks of tryptophan that form. And these collective interactions of light with these networks of tryptophan can produce novel effects, and that's what we show in this work. So, as I said, you can look at a single molecule, and we can treat it, tryptophan in particular, as a two-level system. So we have a molecular resonance, a ground and an excited state, right? And so you put in a photon with the correct amount of energy and you go from the ground to the excited state. Now, if you put two of those tryptophans together, they begin to interact. And so you can get aggregate states, one with a higher frequency and one with a lower frequency. You can call this superradiant and subradiant, upper polaritonic and lower polaritonic. They have many different names in different contexts. But what's important to remember is that that molecular resonance is associated with a strong center frequency, and these aggregate states are shifted, usually red shifted and blue shifted by a certain amount, depending upon the interactions in your network. Now, what happens if we don't just have two molecules, but we have tens of thousands or even millions of these tryptophans in these networks, which we do? Well, even in the single photon limit, so an ultra weak excitation, which you could get even in the background from the quantum vacuum, but you can also, as we've heard the other speakers say, you can get from these ultra weak photon emissions. Even if you put a single photon into your network, you can have a coherent superposition of that single photon excited across the network of N chromophores. So you see it here represented as E being 1 in the excited state, or E being 2 in the excited state, and all others in the ground state, all the way till the last molecule has an excitation and all others are in the ground state. But in quantum mechanics, you can have a coherent superposition of all of those N states, and this is in the single photon limit. So it's fundamentally quantum, and it's been described by Marlon Scully, a laser pioneer, as the super of superradiance. It's also been exploited for quantum information purposes in many other different contexts. So this is the only slide where it's going to be heavy on the math, but it's important to remember that all living systems are open systems. If you put a living system in a closed box forever and it's truly sealed, it's going to die. So in open quantum systems, we're talking about openness to the electromagnetic field. And so there are these tools called non-Hermitian quantum systems that we essentially plug this network of tryptophans into this Hamiltonian, and we can derive all the interactions of this network. So we have a non-Hermitian system that's open to the electromagnetic field. And what happens when you have a non-Hermitian system? You have complex values in your eigenvalues that you get when you solve the Schrodinger equation. So we've solved the Schrodinger equation, we get these energies of our system, and those energies are defined by a Lamb shift, a collective energy shift from what you would get from the single tryptophan resonance, and a collective decay rate that is due to the interactions of the collective mediated by the background electromagnetic field. We've traced out the degrees of freedom of the field so that everything is now represented in terms of the degrees of freedom of our tryptophan network. So what does that look like in microtubules, for example? This is from our 2019 work. You see this beautiful spiral cylinder geometry of the microtubules. And within that, you can see the excited state transition dipoles of each tryptophan. Now, it might not be obvious, but each of those little red arrows corresponds to the 1LA excited or emissive state of tryptophan. Tryptophan is a strongly absorbing, strongly fluorescent molecule with an indole moiety. And so it's of all the amino acids, tryptophan is by far the most fluorescent. So it's used in countless applications for protein folding and monitoring kinetics. Now, we were surprised to see this, but you may not notice that within the microtubule, because of this spiral cylindrical symmetry, you get a buildup of this superradiant state in the lowest excited state of the manifold. So what does that look like? If we plot the collective decay rate, you can think of this as a superradiant enhancement factor of the fluorescence rate. If we plot that on the y-axis, that's the big gamma is the collective decay rate, and little gamma is the single tryptophan decay rate. And we plot that against the energy shift, or the collective Lamb shift. That's the shift due to the quantum vacuum. You'll notice that as we build our microtubule from 1 spiral to 10 spirals to 100 spirals, you see this large increase, orders of magnitude, in the lowest excited state, and that is the signature of superradiance, okay? Now, it might be that, it could have been that the microtubules actually just do this because they're repetitive. So what we did was we randomized the dipoles in the configuration of the microtubule and then just repeated it from 1 spiral over 100 spirals. But we don't get anything close to what we see in the native configuration. So this was a clue to us that, oh, something's going on here that's important and relevant. And it's precisely due to the long-range interactions of the system, not something that we would see if we were only considering just how each tryptophan interacts with each other. So this is what that quantum wave function looks like projected, the superradiant state projected onto the tryptophan site basis, and I'll go more into this later. So here's a close-up picture of those architectures. The centriole is one of the most evolutionarily conserved organelles in all of eukaryotic organisms. The axoneme also is in cilia and flagella. And of course, these are typical axons from mammalian bundles that can scale up to half a micron across. They can have many more than 19 microtubules in each. But each of these tubulin dimers has 8 tryptophans in it. So in a micron microtubule, you have about 10,000 of these tryptophans building up the superradiant state. So I won't go through, but if people are interested in the details, I can answer them later. But the idea is that we can know the probability that the trip network is described by a particular energy eigenstate in thermal equilibrium. It's defined by this equation. We know how to compute the thermally averaged quantum yield by our radiative and non-radiative decay rates. And then because this is an open system, which absorbs photons and then emits them to the electromagnetic field, we can actually define the thermally averaged radiative decay rate over our network of different eigenstates of our Hamiltonian. So this is the thermally populated Gibbs ensemble that gives us in thermal equilibrium. So we're not looking at any fancy non-equilibrium cases. We're choosing the most thermally turbulent environment where it would in principle be hostile to quantum effects. So what we were surprised to find is that that quantum yield actually increases, not only in microtubules, up to 10,000 tryptophans shown here, but in other structures that go to 100,000 tryptophans. And so we're only limited here by the computational scaling, which was really tough for some of these larger structures, which take on the order of months to compute. But you see there's no saturation when you go here to microtubule bundles and centrioles. So now this is experiment. So I was showing you theory, but you'll see experiment in all my slides with red in the upper left, experiment. So here you can see the absorption and fluorescence spectra of tryptophan, tubulin, and microtubules. You'll notice the prominent absorption emission Stokes shift for all 3, but it's much larger in tryptophan because tryptophan in aqueous solution doesn't have the protein environment protecting it, right? So it's much smaller for microtubules and tubulin dimers. Now, there's a lot of details here, but I'm going to highlight the most important part, which is notice that as you go from tryptophan to tubulin, the quantum yield due to the tryptophan molecules alone decreases. And that's important because the non-radiative decay rate is increasing. So you increase the denominator, you decrease the quantum yield, okay? But as you go from tubulin to microtubules, the only thing that's changing is the architecture. Oh no, and now I'm frozen again. Okay, Okay, hopefully, let me try again.
Yeah, you're back. You seem okay.
Okay. Okay, I will share this. Okay. 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 Can you see that okay?
Yes. It looks great.
Okay, let me go back to where I was. So, key point is that the radiative decay rate is increasing and the quantum yield is increasing accordingly. And this is directly due to a superradiant collective quantum optical effect. So this is a really important finding because it's the world's first demonstration of a collective quantum optical effect at micron scale in a solvated protein aggregate at room temperature. So this is happening in a warm environment. It is solvated, but there is still a collective quantum optical effect that is delicate but statistically significant. So how does this scale as we go into larger organelles like centrioles? Well, in fact, for centrioles, the effect is even larger as predicted. There are some challenges with experimentally measuring this, but instead of just hundreds of times as a single microtubule, a centriole is 27 microtubules, and so you can see thousands of times increase in the superradiant effect. And this is actually robust to disorder. So as we increase the disorder from pristine absolute zero centrioles all the way to thermal environments at about 200 inverse centimeters, you still see order of magnitude enhancements due to the superradiance. And that's exciting because biology can use whatever physics is available to enhance transfer. And an order of magnitude enhancement would indeed do things like that, even though it's not necessarily thousands of times more efficient, okay? Now, why is this relevant to quantum yield? Well, we show here that for microtubules and centrioles, this effect is extremely robust and for the quantum yield as a figure of merit. So the quantum yield, even though it's it's a routine observable in many biological labs and photophysics labs, the key point is that because these superradiant states are concentrated in the lowest part of the energy spectrum, that weights this energy even more. The minus of a small value will give you a small exponential. So that weight is very large, and so that weights these superradiant states extremely high. So it appears that biology has found a way to exploit these molecular symmetries in the photoactive degrees of freedom to actually optimize for this quantum yield effect, the proportion of photons that are emitted from those that are absorbed. So we see this also in mammalian neurons, and you can see thousands of times the single tryptophan decay rate, even at a few hundred nanometers. And this extends even larger for bigger bundles. And because of our intensive computational simulations, we've been able now to get really nice analytical models for these scaling. So we can do these simulations on even larger in principle structures. We also see this UV superradiance in locomotive structures like cilia and flagella, but it's much less. And we would expect that because these may be photosensitive, but it's not their primary purpose. They're mechanical in nature. So now, why might this be operative or important in these structures? You've heard about ultra-weak photon emissions. You see that their mitochondria emit these reactive oxygen species and other free radicals that then are electronically excited, and they can emit photons in the ultraviolet spectrum, which can then be picked up and absorbed and used for signaling in these protein aggregate complexes in biology. So when these ultra-weak photon emissions hit a microtubule, imagine you've got, or at 280 nanometers, you've got thousands of these tryptophans within a wavelength of that light. So there's some saturation, of course, as you go to larger and larger system sizes. But what does that look like? Here is that quantum wave function projected onto the tryptophans of about 800 nanometer microtubule. So if you want to get a picture in your mind, see how the probability is mostly localized along the surface of the microtubule rather than on the inner lumen surface. So this is interesting because superradiant states are ultra fast states. Their lifetime is sub-picosecond, okay? So they're faster than thermal noise, and so they would beat some of these thermal effects that are happening on the surface. If you're interested in how they structure, we can actually computationally project that superradiant state onto bundles of smaller microtubules in that structure. And we see the formation of kind of a coherent superposition of superradiant states across those segments of the microtubule. Okay, I'm frozen again, so I'm going to just start sharing from here. So let me play from here. Okay. I'm gonna just show my slides like this, if that's okay, so there's no delay or lag.
Yeah,
okay.
It looks fine from here.
Let's go from here. So this is what a subradiant state.
Hey,
yep.
We saw something for a sec, but now we're not seeing anything on your screen.
You're not seeing it. Okay.
It's a mountain of sand.
Really sorry for these challenges. It's
No
unusual.
problem.
Okay. It says I'm sharing, but I will share again. Okay, can you see now?
Yes.
Okay, so whenever you have a superradiant state, those superradiant states are paired with subradiant states. That's how the math works out. But when you have these subradiant states, these are actually long-lived states. So there's kind of a duality here. You've got superradiant states that are ultra-fast, sub-picosecond, and subradiant states that exist on extremely long time scales. So these are collective quantum states operating at thermal equilibrium that exist for long time frames. And that's really interesting because if you look at this most subradiant state in that 800 nanometer microtubule, it's primarily localized on the inner surface, where you have a highly ordered aqueous environment relative to the environment in thermal equilibrium on the outside. Biology is heterogeneous, as Stuart told us at the very beginning. So we have this heterogeneous environment that biology may be exploiting on the inside and outside of these structures. And there are different processes associated with those superradiant and subradiant states, including supertransfer and subtransfer. And it's not surprising that in quantum information processing, these subradiant states in a very different context that's very cold have been thought of, these so-called dark states have been used for quantum information processing, and there are many protocols to how do we exploit these long-lived states for quantum information processing. So I'm going to go straight to then what is the upshot for brain processing? I know people have some questions about how do these results that we're seeing manifest in how we think of the brain. So because this one's animated, I'm just going to try and share it. Let's go here and let me try to do it from here. Okay. Okay. Can you see it, Paige?
Yes.
Okay, great. So this is a summary of all the different structures that we looked at in this. Oh, my goodness. Okay. Okay, it's not my day today, but I'm going to try to do it this way. Okay. So this is a picture of all those different structures that we see in organelles, in the brain, in axons, axonemes, and in the microtubule. And you can see all these different, in the video, all these different kind of environments that we see in the brain, right? But notice what I said, the sub-picosecond timescales of these superradiant states. These are ultra-fast states that are processing at terahertz frequencies or beyond. Right? Now, when conventional estimates in neuroscience for the lower bound computational speed for the brain go as follows. They say we've got 100 billion neurons, operating at millisecond or kilohertz frequencies. That is the time scale for Hodgkin-Huxley chemical signaling. So that gives us 10 to the 14 ops per second. But if we take 100 billion neurons at terahertz frequencies, right? I'm not saying this is happening continuously, but in a pulsed fashion, we've got 10 orders of magnitude increase in ops per second. 10 to the 23 far exceeds the exascale. That's the frontier of our high performance computing systems. And this is happening in the brain. Now, the energy consumption requirements of the brain, if this is true, cannot scale like a classical digital computer for the following reasons. So Frontier supercomputer, our Frontier HPC system, operates at 10 to the 18 ops per second using 20 megawatts. That's near the power of a nuclear bomb, okay? But the brain at 10 to the 23 ops per second operates on 1/3 the power of a light bulb, okay? So how do we explain the scaling of energy with the vast computational information processing increases that we can see in the brain? And at the same time, notice that you have these ultra fast processing speeds that are paired with long time, second processing speeds, even at tens of seconds in these very, very small axons. Okay, so you have these systems of tryptophans, and I wrote this in the single photon limit, which interestingly enough is very similar. I can rewrite it suggestively as ones and zeros. And this is what's called a W state in quantum computing. And these are very useful because W states in quantum computing are robust to cutting off a qubit or adding a qubit. So if I cut off a qubit here, I still maintain entanglement between the n-1 qubits. And that's important because as you imagine in biology, you're constantly adding and removing tubulins from microtubules, from other polymers that are polymerizing and depolymerizing. And so how do you maintain in a dynamic fashion all these different quantum states? You would use, for example, a W state, especially if you've got a photoexcitation, a weak photoexcitation in your system. So in the time that I have left, hopefully there's some, I just want to present some of the upshots of this. This is a new project funded by the Alfred P. Sloan Foundation, and this is work by our collaborator, Masashi Ono, that he is looking at Physarum polycephalum. It's a unicellular slime mold, an ancient organism that's neither plant, animal, or fungi. And essentially what he had done a few years ago, and it received a lot of press, was to show that the slime mold can actually solve the traveling salesman problem in this optical feedback loop that is controlled by a Hopfield neural network. So here is an 8-city traveling salesman problem. You have n squared lanes, so 64 lanes, that tells you the city and the order in which the city is visited. And you can see here in the body of Physarum, you can see this network of veins that are mainly actomyosin tubes, but also contain some microtubules. And you'll notice that you can see in the thickness plots here, this beautiful kind of coherent oscillation that you're seeing. It remains to be seen whether this is quantum, but what's interesting is that as you advance this slide and you keep stimulating this optical feedback loop that's controlled by a Hopfield network, you get a beautiful solution to the traveling salesman problem that is high quality. So this is an NP-hard problem that, at least in this instance, is actually solved in linear time as you scale with the number of cities. I haven't shown you the smaller city sizes, but up to 8 cities, we can solve an NP-hard problem in linear time using this setup. And so we're rebuilding this setup, and we hope to go to even larger city sizes and to say, can we harness potentially quantum effects in a small organism to actually produce the solution to an NP-hard problem? Okay. And in addition to that, we're looking experimentally with Nirosha Murugan and others in the Tufts lab at mass sensing paradigms, novel imaging of the slime mold, and what is the smallest fundamental unit that allows these slime molds to reintegrate after they've been kind of eviscerated into little parts. Okay, For those of you who are consciousness on the philosophy side, experts or people who are interested, I did want to, without going into too much detail, make the important point that these physical laws and equations that we're using, like Newton's and Schrodinger's and other physical laws, they do not necessitate all of these structures that we're describing, right? Physics doesn't tell me that there should be life, brains, water, ecosystems, or anything else in that we observe in the physical universe, because we are sufficiently ignorant of universal initial conditions. And I mean of the universe when I say universal. So the classic example was from C.D. Broad. It's an old work cited by Dave Chalmers from the early 20th century. And he considers a mathematical archangel, a superintelligence. Would that superintelligence be able to derive the smell of ammonia or the slipperiness of water merely from the chemical structure? And it makes the important point that even if I give you all the eigenvalues, even if I'm able to compute all of those eigenvalues, there's something still missing. There's a gap, an epiphenomenal or phenomenal gap between physical laws and observables in science and what we experience as conscious agents of processing. So I'll leave that concept and skip over this in the interest of time. But to summarize and conclude, this is the standard model of biology. It's what you're taught in high school. It's what you're often taught in college. And it involves things like assumptions of thermal equilibrium, linear response and ergodicity, things I didn't talk about today, like Markovian dynamics and the Born-Oppenheimer approximation, where you can treat electrons and protons essentially as separated in scale. But what we're showing in my group and in many others is that these assumptions are actually not valid in living systems and that we have to treat them more carefully. And so these works, many from my group, but also from a few others, show that this is really an emerging paradigm that we need to take seriously. So the Quantum Biology Lab at Howard University in Washington, D.C. sits at the nexus of theoretical physics, applied math, and biological and chemical physics. We investigate everything from quantum foundations to quantum electrodynamics and neuroscience. And we have many different projects and are looking for people from undergraduate to postdoc levels to join us. So I will conclude there and leave you with this quote from one of the founders of quantum theory, Max Planck, who reminds us that all the science that we're doing brings news of another world which lies outside of ours and is entirely independent of us. So thank you so much for your time.
And thank you, Philip. That was fantastic. That was very exciting, and to hear about the work you're doing there. And I'd love to, actually, being based in Maryland myself, I'd love to come take a visit out there and see
Absolutely.
some of it for myself. So we have time for, well, we don't, but we're going to take a couple questions. Is there any questions from the audience? Yes.
Thank you very much for the intriguing talk and the rate in which you talk. I'm curious.
Thank you.
I'm curious. What is your imagination and creativity style? How do you go about accessing everything that you talk about logically? How do you access that yourself and bring it forth?
That is a very complex question. It's without method and quite often inexplicable. But I will say that I do consider myself an interactionist. So in the sense that a type D dualism is my way of thinking about the interaction between physical objects, which I include myself as part physical, and things that exist beyond. So the creative aspect, that which comes from beyond, just like in the physical world, is an interaction between light and life. So if you interact with light, you are interacting with that which lies beyond in the quantum vacuum. And so how you get there that we need to have a coffee or a beer.
Let's do that, because the concept of how we access light, okay, if you look at the history of many cultures across the planet, you'll find many of them talking about there's an inner light. How do you access, how do you do that? And so one of the things I'm hearing here from this program is now there's more science around these internal light structures in the body, and my work is how do you scale that? So my name's Dan Lapp, and I'm part of NASA's Astrobiology Science Communication Guild. So I'm looking at public science communication. How do we help people with this? You know, public health, education, sports, relationship, mental health, et cetera. How do you take what we have here and help people access that light and make it simple, make it practical every day? Got your contact information. You seem like a great guy. Let's do that herbal tea sometime, okay?
Okay, great. Thank you.
All right, great. So thank you so much, Philip, and also I want to thank all of the speakers today. This was a fantastic and exciting session, and I wish we had a little more time to get into that panel discussion we were planning, but we ran a little long today. So thank you very, very much for joining us. It was really, really exciting.